The ocean has always beckoned. Throughout the vast expanse of evolutionary history, spanning hundreds of millions of years, countless lineages of terrestrial vertebrates have abandoned the security of solid ground and ventured back into the aquatic realm from which their distant ancestors originally emerged. This phenomenon, known as secondary aquatic adaptation, represents one of the most remarkable transformations in the natural world. At least 7 major groups of mammals independently evolved to exploit marine environments, each following their own unique pathway from land to sea. These extraordinary transitions provide us with a blueprint, a roadmap of biological possibilities that illuminates what might happen if humans were to follow the same path and return to the ocean. The story of mammals returning to water is not a tale of recent origin. The fossil record reveals that these transitions began remarkably early in mammalian history, with some lineages making their aquatic journey more than 50 million years ago. The most spectacular and well-documented of these transformations belongs to the cetaceans, the group that includes modern whales, dolphins, and porpoises. Their evolutionary journey from four-legged, furry land mammals to the sleek, streamlined oceanic giants we recognize today took approximately 8 to 10 million years. This timeframe provides a crucial benchmark for understanding the scale of adaptation required for a complete transition from terrestrial to fully aquatic life. The cetacean story begins in the shallow coastal waters and river deltas of the ancient Tethys Sea, a vast ocean that once separated the supercontinents during the Eocene epoch, approximately 55 to 35 million years ago. During this time, the planet was significantly warmer than it is today, with tropical and subtropical conditions extending far beyond their current ranges. The Tethys Sea provided an ideal environment for the evolution and diversification of early whales, with productive shallow waters rich in fish and other prey species. It was in the sediments deposited along the margins of this ancient ocean, in what is now modern-day Pakistan and India, that paleontologists would later uncover the fossilized remains of creatures that would revolutionize our understanding of whale evolution. Before we can discuss the whales themselves, we must first understand their unlikely ancestor. In 1971, Indian geologist A. Ranga Rao discovered several unremarkable fossils in the rocky landscape of Kashmir, comprising a few teeth and a jawbone fragment. These fossils remained in relative obscurity for decades, their true significance hidden from scientific scrutiny. It was not until Dr. Hans de Wissen examined these specimens, donated by Rao's widow after his death in 1999, that the fascinating story of Indoeus began to emerge. When a technician accidentally broke open a skull from Rao's collection, they revealed an extraordinary structure, a robust ear bone known as an involucrum. This specialized bone had previously been observed only in whales and other cetaceans, where it facilitates underwater hearing. Indoeus, named after India and the Greek word for water pig, was a small, raccoon-sized mammal that lived approximately 48 million years ago during the Eocene epoch. This chevrotain-like creature, resembling a modern mouse deer, had a long snout, a lengthy tail, and hooves on its feet. Despite its terrestrial appearance, Indoeus possessed anatomical features that revealed its connection to water. Beyond the involucrum, this animal had exceptionally dense leg bones, a characteristic seen in modern hippopotamuses. Hippos utilize their heavy bones to remain submerged while walking along river bottoms, capable of holding their breath for up to 6 minutes when necessary. The combination of aquatic hearing adaptations and dense bones strongly suggests that Indoeus inhabited both terrestrial and aquatic environments, likely spending significant time in water to forage for food or escape from predators. The discovery of Indoeus in the high Himalayas, far from any ocean, might seem paradoxical. However, during the Eocene epoch, this region was part of a coastal environment along the northern edge of the Tethys Sea. The geological forces that would eventually thrust these sediments upward to form the Himalayan mountain range had not yet begun their work. Instead, these areas were low-lying river deltas and coastal plains, perfect habitats for semi-aquatic mammals making their first tentative steps into an aquatic lifestyle. From creatures like Indoeus, the evolutionary lineage leading to modern whales began its remarkable transformation. The next significant fossil in this sequence is Pachycetus, whose first remains were discovered in 1978 by paleontologist Phil Gingerich along the shores of the ancient Tethys Sea. The species was formally named and unveiled to the scientific community in 1981, though the initial discovery consisted only of a skull and jaw fragments. These bones alone were sufficient to reveal that Pachycetus was a land-dwelling animal, with key features identifying it as an early whale. Additional fossils discovered in the Kuldana Formation west of Islamabad in northern Pakistan provided a more complete picture of this remarkable creature. Pachycetus inachus, the best-known species of the genus, lived approximately 48.5 to 50 million years ago in river delta deposits along the northern edge of the Tethys Sea. This animal was roughly the size of a large dog or small wolf, with 4 functional legs capable of locomotion on land. Unlike modern whales, Pachycetus retained many features of terrestrial mammals, including an auditory system better suited for hearing in air than in water. However, critical anatomical details revealed its place on the pathway to fully aquatic life. The structure near its ears showed distinct similarities to modern whales, and its teeth were distinctive, differing from typical terrestrial carnivores while showing characteristics that would become more pronounced in later cetaceans. The teeth of Pachycetus provide particularly valuable information about its lifestyle and diet. Multiple broken teeth and skull fragments found in the fossil deposits suggest that Pachycetus was a predator, likely hunting fish in shallow freshwater environments using an ambush strategy. Its dentition was not unlike that of its closest terrestrial relatives, including creatures called mesonychids, but showed modifications consistent with an increasingly aquatic lifestyle. The discovery of various skeletal remains has revealed that multiple types of Pachycetus coexisted, with different species identified based on their unique dental features. One of the most significant anatomical features linking Pachycetus to later whales involves the positioning of a particular joint in the skull. In terrestrial mammals, this joint is oriented in a specific direction, but in modern cetaceans, it has rotated to a completely different position. Pachycetus represents an intermediate condition, with the joint oriented rostrally, positioned between the ancestral terrestrial state and the derived aquatic condition. This clear rotation from the ancestral condition provides a snapshot of the transition taking place. The environment in which Pachycetus lived played a crucial role in its evolution. The fossil deposits containing Pachycetus remains include assortments of limestone, dolomite, stone mud, and various colored sands, predicted to be a favorable habitat for this early whale. During the Eocene, the region that is now Pakistan was part of an independent island continent off the coastal region of Eurasia, providing an ideal habitat for the evolution and diversification of early whale species. Pachycetus occurred alongside a land mammal fauna in fluvial sediments bordering epicontinental Eocene remnants of the eastern Tethys Seaway. This discovery strengthened inferences that whales originated from terrestrial carnivorous mammals and suggested that they made a gradual transition from land to sea in the early Eocene. The evolutionary hypothesis suggests that these early whales spent progressively more time feeding on planktivorous fishes in shallow, highly productive seas and embayments associated with the tectonic closure of eastern Tethys. This gradual transition meant that each generation spent slightly more time in water, developed slightly better swimming abilities, and became incrementally more adapted to aquatic prey capture. The selective pressures favoring these adaptations were likely related to food availability and competition. Coastal waters rich in fish would have provided abundant resources for any predator capable of exploiting them effectively. The next major milestone in whale evolution is represented by Ambulocetus natans, whose name translates to walking whale that swims. This remarkable creature lived approximately 49 million years ago in what is now northern Pakistan, in long-lost coastal shallow seas and brackish rivers. Ambulocetus represents a critical intermediate form, possessing anatomical features that allowed it to function both on land and in water. Unlike modern cetaceans, which are entirely helpless on land, Ambulocetus was certainly capable of walking on land, probably in a manner similar to modern sea lions or fur seals. The lifestyle of Ambulocetus can be inferred from its skeletal structure and the environment in which its fossils are found. This animal was considerably larger than Pachycetus, with a more robust body plan adapted for swimming while retaining functional limbs for terrestrial locomotion. Its name reflects the dual nature of its existence, straddling the boundary between land and sea. Fossil evidence suggests that Ambulocetus was a formidable predator, capable of hunting both in water and on land, though increasingly specialized for aquatic prey capture. As whale evolution continued through the Middle Eocene, approximately 46 to 40 million years ago, species like Rhodhostis emerged, pushing the aquatic adaptations even further while still retaining several primitive terrestrial features. Rhodhostis is particularly significant because it is the earliest archaeocete for which all of the thoracic, lumbar, and sacral vertebrae have been preserved. These vertebrae reveal important information about locomotion. The lumbar vertebrae had higher neural spines than in earlier whales, indicating attachment points for larger muscles. The size of these extensions on the top of the vertebrae, where muscles are attached, indicates that Rhodhostis had developed a tail fluke, though such soft tissue structures are not preserved in the known fossils. Several anatomical features support the interpretation that Rhodhostis possessed functional tail flukes: shortened cervical vertebrae, heavy and robust proximal tail vertebrae, and large dorsal spines on the lumbar vertebrae for large tail and other axial muscle attachments. These features are associated in modern whales with the development and use of tail flukes. All evidence suggests that Rhodhostis was a very good tail swimmer and represents the earliest fossil whale committed to this manner of swimming. The pelvis of Rhodhostis was smaller than that of its predecessors, but it was still connected to the sacral vertebrae, meaning that Rhodhostis could still walk on land to some degree. However, the ilium of the pelvis was short compared to that of the land-dwelling mesonychids, making for a less powerful muscular thrust from the hip during walking. The femur was about one-third shorter than in earlier species, further indicating reduced terrestrial capability. Rhodhostis represents a creature increasingly committed to aquatic life, but not yet entirely divorced from the land. The skull of Rhodhostis was rather large compared to the rest of the skeleton, with the premaxillae and dentaries extending forward even more than its predecessors, elongating the skull and making it more cetacean in appearance. The molars had higher crowns than in earlier whales and were greatly simplified. The lower molars were higher than they were wide, a dental pattern associated with fish eating. Environmental evidence from the fossil deposits provides additional insight into the lifestyle of Rhodhostis. This species is found in green shales deposited in the deep neritic zone, equivalent to the outer part of the continental shelf. Because green shales are associated with fairly low oxygen bottom waters, Rhodhostis must have lived at greater water depths than any previous cetacean. The fact that it is found in association with planktonic foraminiferans and other microfossils agrees with this determination of water depth. The late Eocene, approximately 40 to 34 million years ago, saw the appearance of Basilosaurus, often called the king lizard despite being a mammal. This name resulted from early confusion about the nature of the fossils when they were first discovered. Basilosaurus was an impressively large whale, measuring 15 to 18 meters in length, with a long, snake-like or eel-like body. 3 species of Basilosaurus are known from fossil sites in the southeastern United States, England, Egypt, Jordan, and Pakistan, indicating that this genus inhabited the northern Atlantic Ocean, Tethys Sea, and the Paratethys Sea, which was the precursor to the Mediterranean Sea. Basilosaurus lived during the Eocene epoch, specifically the Pre-Devonian stage, approximately 37.8 to 33.9 million years ago. This creature represents a critical evolutionary grade in whale evolution, showing advanced aquatic adaptations while retaining some primitive features. Contemporary with Basilosaurus was Dorudon, a smaller whale that probably represents the group most likely to be ancestral to modern whales. Dorudans swam the ancient Eocene oceans of what is now Egypt and the United States alongside Basilosaurus. The early Oligocene period that followed is thought to be when odontocetes, the toothed whales, and mysticetes, the baleen whales, diverged into separate evolutionary lineages. The transformation from Pachycetus to modern whales represents one of the most dramatic morphological changes documented in the fossil record, occurring over approximately 8 to 10 million years. During this time, these mammals transitioned from four-legged, furry land animals to completely aquatic, streamlined forms. The fossil sequence from Pakistan and other Tethys margin sites provides an exceptionally complete record of this transformation, with numerous intermediate forms filling in the evolutionary steps. Cetaceans were not the only mammals to make this journey back to the sea. Pinnipeds, the group including seals, sea lions, and walruses, represent another independent aquatic transition. The earliest known pinniped is Echolocation, which lived during the early Miocene, approximately 23 to 20 million years ago. Echolocation was described in 1973 from the Pyramid Hill member of the Jewett Sand in Kern County, California. This early pinniped still bore an ursid-like or bear-like shearing dentition, which is absent in all modern pinnipeds. Modern pinnipeds have generally homodont postcanine teeth, meaning the teeth behind the canines are all similar in shape. Echolocation is considered basal to all modern pinnipeds, representing a common ancestor from which seals, sea lions, and walruses descended. The fossil evidence suggests that pinnipeds derived from a bear-like ancestor sometime during the late Oligocene, probably in the Northeast Pacific, where the oldest pinniped fossils are known. However, pinniped fossils seem to simply appear in the fossil record after sea levels rose and deposited more marine sediments during the late Oligocene in both the Pacific and Atlantic Oceans. This pattern suggests that even more primitive pinnipeds may be found in earlier sediments or as Lazarus taxa in late Oligocene rocks. The pinniped transition occurred approximately 36 million years ago, making it considerably younger than the initial cetacean transition, but still representing tens of millions of years of evolutionary change. Pinnipeds retain their connection to land much more thoroughly than cetaceans, continuing to haul out on beaches and ice to rest, breed, and give birth. This semi-aquatic lifestyle represents a different evolutionary pathway, showing that complete independence from land is not inevitable for marine mammals. A third major group of aquatic mammals, the sirenians, took yet another path. Modern sirenians, including manatees and dugongs, are the only extant herbivorous marine mammals and the only group of herbivorous mammals to have become completely aquatic. Sirenians were among the first mammals to retransition from land to water and hold the distinction of being the sole herbivorous marine mammals. The ancestors of extant manatees and the dugong diverged at Crown Sirenia approximately 30 million years ago. The earliest known fully quadrupedal sirenian possessed complete hindlimbs, but these were gradually lost as the lineage became more aquatic. The sirenian evolutionary path reveals unique adaptations. Unlike cetaceans and pinnipeds, which are carnivorous, sirenians evolved to exploit aquatic plant resources. This dietary specialization required distinct digestive adaptations and foraging behaviors. Genetic studies have revealed convergent regression of integumentary system genes shared with cetaceans, indicating that multiple independent lineages solved similar problems, such as hair loss and skin modifications, through parallel genetic changes. Additionally, sirenians developed tolerance to a high-iodine plant diet mediated through changes in the iodide transporter and its co-transporters. The phenomenon of convergent evolution becomes particularly striking when comparing these independent marine transitions. Despite arising from different terrestrial ancestors at different times and in different locations, cetaceans, pinnipeds, and sirenians all evolved remarkably similar body plans. This morphological convergence includes a streamlined body shape with modified limbs. Pinnipeds have front and hind limbs modified as flippers, whereas cetaceans and sirenians completely lost their hind limbs. Most marine mammals present respiratory systems adapted for reduced oxygen consumption, which enables them to withstand hypoxia and prolong deep dives. Recent research has identified convergent evolution at multiple biological scales, from anatomical structures down to genetic sequences. The repeated evolution of hydrodynamic limbs and axial modifications likely exploited parallel developmental mechanisms. Such shared pathways may extend to the level of gene regulation, linking genomic and phenotypic convergence. This means that different lineages independently evolved similar body forms, not just because they faced similar environmental pressures, but because they had access to similar genetic and developmental toolkits that could be modified in predictable ways. A recent Yale study examining why evolution repeatedly returned animals to water found that the transition from terrestrial to aquatic life in tetrapods has occurred at least 34 separate times over the past 250 million years. This remarkable pattern suggests that aquatic environments offer significant advantages that repeatedly attract terrestrial lineages. The study found that form proportion changes could predict flipper development with 90% accuracy, indicating that there are consistent, predictable pathways that evolution follows when adapting limbs for swimming. The success of these multiple independent transitions demonstrates that returning to the ocean is not only possible, but, under the right circumstances, highly advantageous. Each lineage found its own ecological niche, its own way of making a living in the aquatic realm. Cetaceans became pursuit predators and filter feeders, pinnipeds remained semi-aquatic ambush hunters and foragers, and sirenians became the only fully aquatic herbivores. This diversity of outcomes shows that there is not one single way to be an aquatic mammal. The ocean offers multiple ecological opportunities for species capable of adapting to its challenges. The stepping stone hypothesis suggests that freshwater environments often served as intermediate habitats during these transitions. Many early whales, including Pachycetus and Ambulocetus, are found in river delta and coastal environments that would have included both freshwater and brackish water habitats. This gradual transition from rivers to estuaries to coastal seas and finally to open ocean allowed for stepwise adaptation, with each generation needing to make only modest adjustments rather than dramatic leaps. Understanding these precedents is crucial for envisioning what would happen if humans followed a similar path. The fossil record provides a detailed roadmap showing which adaptations appear first, which take longer to develop, and how different organ systems are modified in concert. The timescales involved, measured in millions of years and hundreds of thousands of generations, make it clear that this would not be a rapid transformation. However, the repeated success of this evolutionary strategy across multiple mammalian lineages demonstrates that the transition from land to sea is achievable. The question is not whether mammals can return to the ocean. We know they can, but rather what specific changes would be required for humans to make that same journey. Chapter 2: The Aquatic Ape Hypothesis, Controversial Foundation. Before examining the realistic evolutionary changes required for humans to become aquatic, we must address one of the most controversial and widely debated ideas in human evolutionary theory: the aquatic ape hypothesis. This hypothesis, while rejected by mainstream anthropology and classified as pseudoscience by the scientific establishment, raises intriguing questions about human anatomy and our relationship with water. Understanding both the arguments of this hypothesis and the reasons for its rejection provides essential context for considering what genuine aquatic adaptation in humans would actually entail. The aquatic ape hypothesis originated in 1960 with British marine biologist Alistair Hardy. Hardy proposed that many distinctive human characteristics, which set us apart from other primates and terrestrial mammals, might be explained by a period in our evolutionary past when our ancestors lived a semi-aquatic lifestyle. He suggested that at some point, perhaps 5 to 7 million years ago, a population of ancestral apes was forced by environmental pressures to exploit aquatic food resources along coastlines, rivers, or lake shores. According to Hardy's proposal, this aquatic phase lasted long enough to leave permanent marks on human anatomy and physiology. Hardy's initial paper was published in the New Scientist magazine rather than a peer-reviewed scientific journal, which limited its acceptance within the academic community. However, the hypothesis gained substantial public attention decades later when Welsh writer Elaine Morgan championed it, beginning in the 1970s. Morgan wrote several popular books on the subject, including The Descent of Woman in 1972 and The Aquatic Ape in 1982, arguing passionately for the aquatic phase in human evolution. Her work brought the hypothesis to a wide audience and sparked ongoing debate that continues to this day. The core arguments of the aquatic ape hypothesis center on several human anatomical features that proponents claim are atypical of terrestrial mammals, but common in aquatic or semi-aquatic ones. The most frequently cited features include our relative hairlessness compared to other primates, the layer of subcutaneous fat beneath our skin, our bipedal locomotion, our descended larynx, voluntary breath control, and various aspects of our reproductive physiology. Proponents argue that these features make more sense as adaptations to an aquatic environment than as adaptations to life on the African savanna, which is the setting for the prevailing model of human evolution. The hairlessness argument points out that humans have far less body hair than any other primate species. While we have roughly the same number of hair follicles as chimpanzees, our body hair is much finer and shorter, creating the appearance of naked skin. Proponents of the aquatic ape hypothesis note that hair loss is common among aquatic mammals, including cetaceans, sirenians, and to a lesser extent, pinnipeds. The suggestion is that our ancestors lost their fur because it creates drag in water, and because aquatic mammals typically rely on blubber rather than fur for insulation. The subcutaneous fat observation forms another pillar of the hypothesis. Humans have a continuous layer of fat directly beneath the skin, distributed relatively evenly across the body. This pattern is unusual among primates and terrestrial mammals generally, but it is characteristic of aquatic mammals. In marine mammals, this blubber layer serves multiple functions: insulation in cold water, energy storage for long migrations, and buoyancy control. Proponents argue that the human subcutaneous fat layer suggests an aquatic phase in our evolution when such insulation would have been advantageous. The bipedalism connection to water is perhaps one of the more interesting aspects of the hypothesis. Hardy and Morgan suggested that upright posture might have originated as an adaptation for wadding in shallow water. When wadding, an upright stance allows an animal to keep its head above water while moving through deeper areas. This would have been particularly useful for early hominins exploiting coastal or riverside resources. The hypothesis proposes that bipedalism first evolved in the water and was later adapted for terrestrial locomotion, rather than evolving on land and then being useful in water. Research on bipedalism evolution has explored various environmental contexts. While the aquatic hypothesis is not accepted by mainstream paleoanthropology, studies have examined the role of hydrostatic forces and water environments in locomotion. Some researchers have noted that upright wadding in water reduces the energetic cost of movement compared to quadrupedal wadding, which could have provided selective advantages for hominins frequenting aquatic environments. However, most evidence points to terrestrial advantages of bipedalism, such as improved long-distance travel efficiency, freeing the hands for tool use and carrying, and better thermoregulation in hot, open environments. Voluntary breath control represents another feature that aquatic ape proponents highlight. Unlike most mammals, which breathe automatically and have difficulty voluntarily holding their breath, humans can consciously control their breathing. This ability allows us to hold our breath for extended periods, with trained individuals achieving breath holds measured in minutes. Aquatic mammals also possess enhanced voluntary control over breathing, which is essential for diving. Proponents suggest that human breath control evolved during an aquatic phase and later proved useful for speech production. The descended larynx in humans is unique among primates and creates an elongated vocal tract that enables complex speech. However, it also makes humans uniquely vulnerable to choking, as our airway and food passage cross in the throat. Aquatic ape proponents note that marine mammals also have descended larynxes, which help create watertight seals during diving and prevent water from entering the lungs. They suggest that the human descended larynx originally evolved for aquatic purposes and was later co-opted for speech. Despite these intriguing anatomical observations, the mainstream scientific community has overwhelmingly rejected the aquatic ape hypothesis. The hypothesis is classified as pseudoscience by anthropologists and evolutionary biologists for several fundamental reasons. First, there is a complete absence of fossil evidence supporting an aquatic phase in human evolution. The hominin fossil record, while incomplete, shows a clear pattern of adaptation to terrestrial and sometimes woodland or savanna environments, with no anatomical features diagnostic of semi-aquatic specialization found in the fossil specimens. Anthropologists point out that every feature cited by aquatic ape proponents can be explained more parsimoniously by terrestrial adaptations. Human hairlessness, for instance, is better explained as a thermoregulatory adaptation for endurance running and heat dissipation in hot climates. The ability to sweat profusely through our relatively hairless skin allows for efficient evaporative cooling, which would have been crucial for persistence hunting on the African savanna. In contrast, aquatic mammals that have lost their hair rely on blubber for insulation because water conducts heat much more rapidly than air. Humans lack the thick blubber layer characteristic of truly aquatic mammals. Our subcutaneous fat is relatively thin compared to seals or cetaceans. The subcutaneous fat pattern in humans, while notable, is not equivalent to marine mammal blubber. Human body fat serves primarily as energy storage rather than insulation, and its distribution varies considerably with sex, age, diet, and genetics. The amount of subcutaneous fat in humans would be inadequate for extended time in cold water, as anyone who has experienced cold water swimming can attest. Studies on thermoregulation show that humans lose heat rapidly in water at temperatures below 35 to 36 degrees Celsius, which is far warmer than most ocean. Regarding bipedalism, the extensive fossil and biomechanical evidence points to terrestrial origins. The earliest hominins showing evidence of bipedalism, such as Sahelanthropus tichadensis, Aurorin tugenensis, and Ardipithecus ramidus, are found in woodland and savanna environments, not coastal or aquatic settings. The anatomical specializations for bipedalism in these fossils, including changes to the pelvis, femur, knee, and foot, are consistent with terrestrial walking and running, not wadding. Additionally, the energetic advantages of bipedalism are most pronounced during terrestrial locomotion, particularly for long-distance travel. The voluntary breath control argument also faces significant challenges. While humans do have enhanced voluntary control over breathing compared to most mammals, this ability is not unique to aquatic species. Many terrestrial mammals, including other primates, can hold their breath to varying degrees. The human capacity for breath control is better explained as a consequence of the neural connections required for speech and vocal communication. The ability to precisely control airflow and timing is essential for producing the rapid, modulated sounds of human language. Speech likely provided the primary selective pressure for enhanced breath control, with any aquatic applications being secondary benefits. Critics also point out that the aquatic ape hypothesis relies heavily on superficial anatomical comparisons while ignoring the many ways in which humans differ dramatically from aquatic mammals. True aquatic mammals show consistent suites of adaptations that humans entirely lack. These include streamlined body shapes, modified limbs functioning as flippers or paddles, nostrils positioned on top of the head or specialized for closing underwater, thick blubber layers, modifications to the cardiovascular system for prolonged diving, eyes adapted for underwater vision, and reproductive systems modified for aquatic birth. Humans possess none of these specialized aquatic adaptations. Our body shape is not streamlined. Our limbs function well for terrestrial locomotion, but poorly for efficient swimming compared to pinnipeds or cetaceans. Our nostrils remain in the standard mammalian position. We lack significant blubber. We cannot dive for extended periods without training and equipment. Our eyes function poorly underwater without goggles, and human births are adapted for terrestrial environments, with infants unable to swim competently and vulnerable to drowning. Furthermore, the proposed timeline for an aquatic phase in human evolution is problematic. For aquatic adaptations to leave permanent anatomical marks on a lineage, the aquatic phase would need to persist for hundreds of thousands to millions of years, as evidenced by the cetacean fossil record showing 8 to 10 million years of transition. However, the entire hominin fossil record from the split with chimpanzees approximately 6 to 7 million years ago shows continuous terrestrial adaptation, with no gap that could accommodate a prolonged aquatic phase. Despite the scientific rejection of the aquatic ape hypothesis as a comprehensive explanation for human evolution, some researchers have proposed more moderate waterside hypotheses that acknowledge humans may have exploited aquatic resources without undergoing specialized aquatic adaptation. These hypotheses suggest that coastal foraging and shellfish gathering may have played important roles in human evolution, particularly during certain periods and in certain regions. Archaeological evidence supports the idea that coastal and riverside resources were important to some human populations. Shell middens, accumulations of discarded shellfish remains, document intensive exploitation of marine and freshwater resources dating back at least 165,000 years. During the Ice Age, particularly around 40,000 years ago, coastal lifestyles may have been crucial for human survival in some regions. Shellfish and fish provided reliable protein sources and, importantly, essential fatty acids, including omega-3 fatty acids and docosahexaenoic acid, that are critical for brain development. The debate about whether access to aquatic foods, particularly those rich in docosahexaenoic acid and other long-chain polyunsaturated fatty acids, was necessary for the evolution of the large human brain remains active. Some researchers argue that marine food chains were the primary source of these essential compounds, and that access to aquatic resources enabled human brain expansion. Others contend that terrestrial food sources could have provided adequate nutrition for brain development. This discussion is separate from the aquatic ape hypothesis itself, focusing instead on dietary ecology rather than anatomical adaptation to an aquatic environment. The distinction between exploiting aquatic resources and evolving aquatic adaptations is crucial. Many terrestrial animals, including various primates, exploit aquatic resources opportunistically without developing specialized aquatic adaptations. Japanese macaques dive for seaweed, chimpanzees fish for algae with sticks, and numerous primate species include aquatic plants, fish, or crustaceans in their diets. Exploiting water-based resources does not require or necessarily lead to aquatic specialization. Semi-aquatic behavior in human ancestors, such as wadding for shellfish or fishing in shallow water, could have occurred without leaving permanent anatomical signatures characteristic of aquatic adaptation. Wadding and swimming behaviors can be performed by animals with generalized terrestrial anatomy. Humans are actually reasonably competent swimmers compared to many terrestrial mammals, partly because our upright posture and mobile shoulders allow for effective arm stroking, and partly because our subcutaneous fat provides some buoyancy. However, this competence does not approach the efficiency seen in animals with genuine aquatic adaptations. What can be learned from the aquatic ape hypothesis despite its rejection by mainstream science? The hypothesis highlights several human traits that are indeed unusual and worthy of explanation. Even if the aquatic explanation is incorrect, the questions raised about hairlessness, fat distribution, bipedalism, and breath control remain valid and have stimulated productive research into alternative explanations. The hypothesis also demonstrates the importance of rigorous hypothesis testing and the danger of superficial anatomical comparisons without considering the full suite of traits and the fossil evidence. Additionally, public interest in the aquatic ape hypothesis reflects a genuine fascination with human origins and our relationship with water. Humans have a complex relationship with aquatic environments. We cannot breathe underwater, yet we are drawn to water for recreation, transportation, and resources. We are not aquatic, yet we are better adapted to water than most terrestrial mammals. Understanding this intermediate status helps frame the question of what would be required for humans to genuinely transition to an aquatic lifestyle. If humans were to actually evolve aquatic adaptations, as opposed to simply exploiting coastal resources, the changes required would be far more extensive than those proposed by the aquatic ape hypothesis. True aquatic adaptation would involve fundamental restructuring of multiple body systems, as evidenced by the transitions documented in the fossil record of cetaceans, pinnipeds, and sirenians. The anatomical features, sometimes attributed to a hypothetical aquatic phase in our past, would represent only the most preliminary steps in a transformation that would take millions of years to complete. The rejection of the aquatic ape hypothesis by the scientific community should not be misunderstood as a rejection of the possibility that mammals, including human descendants, could evolve aquatic adaptations. The fossil record unequivocally demonstrates that such transitions have occurred repeatedly. The rejection specifically addresses the claim that humans have already undergone such a transition in our evolutionary past. The evidence does not support that claim. However, the evidence does support that future human evolution in an aquatic direction would be theoretically possible, given sufficient time, appropriate selective pressures, and genetic variation for selection to act upon. Understanding both the appeal and the flaws of the aquatic ape hypothesis provides valuable context for examining what realistic aquatic evolution in humans would actually entail. It clarifies which human traits are genuinely pre-adaptations that might facilitate an aquatic transition and which would need to be developed from scratch. It also establishes the scope of changes required and the timescales involved, using the well-documented transitions of other mammalian lineages as guides. Chapter 3: Current Human Aquatic Capabilities, our starting point. Understanding what happens when humans actually evolve toward aquatic life requires first establishing where we currently stand in terms of aquatic capabilities. Despite being terrestrial mammals, humans possess a surprising array of physiological responses and adaptations that function when we enter water. These existing capabilities represent the foundation upon which any future aquatic evolution would build. Some of these traits are shared with all mammals, while others show intriguing variations that hint at the potential for further development. The most fundamental aquatic response in humans is the mammalian diving reflex, also known as the diving response, which exists in all mammals including humans. This ancient physiological mechanism is hypothesized to aid in preserving oxygen stores for key organ systems during breath-hold diving. The diving response represents a coordinated series of automatic adjustments that occur when the face, particularly the area around the nose and eyes, comes into contact with cold water. These adjustments happen without conscious control and demonstrate that our bodies retain ancient programming for aquatic survival. The primary components of the mammalian diving reflex include bradycardia, which is the automatic slowing of heart rate, peripheral vasoconstriction, which involves the narrowing of blood vessels in the extremities, and blood shift, where blood redistributes from the limbs to the vital organs. When a human face becomes submerged in cold water, the heart rate immediately begins to decrease. This bradycardic response is mediated by the vagus nerve and represents a parasympathetic nervous system activation. The colder the water, the more pronounced the response becomes, with some individuals experiencing heart rate reductions of 20 to 30% or more. Peripheral vasoconstriction occurs simultaneously with bradycardia during submersion by resistance vessels limiting blood flow to muscles, skin, and viscera, which are regions considered hypoxia-tolerant. This redirection preserves oxygenated blood for the heart, lungs, and brain, the organs most sensitive to oxygen deprivation. The narrowing of blood vessels to reduce blood flow happens through muscular contraction of the blood vessel walls. This selective reduction in circulation to non-essential areas allows the limited oxygen supply to be concentrated where it is most critically needed. Blood shift represents the third major component of the diving response. Upon diving, atmospheric pressure increases with depth where, according to Boyle's law, the lungs and the air contained inside of them will compress as the free diver descends beneath the surface. Due to the peripheral vasoconstriction initiated by the diving reflex, blood will be shunted from the extremities into the vital organs and thoracic cavity, resulting in a higher percentage of blood volume in this area that will occupy the space created by the compression of air inside the lungs. This blood shift serves a protective function, preventing lung collapse at depth by filling the thoracic cavity with incompressible fluid. The diving response provides immediate benefits even for untrained individuals. However, the strength and efficiency of this reflex can be dramatically enhanced through training and conditioning. Modern freedivers have pushed the boundaries of human breath-hold capabilities to extraordinary lengths. In 2025, Croatian freediver Vitomir Maričić set a new world record by holding his breath underwater for 29 minutes and 9 seconds. This achievement eclipsed the previous record of 24 minutes and 37 seconds, set by Budimir Šobat in 2021. These times are extraordinary when compared to the average untrained human, who can typically hold their breath for only 30 to 90 seconds. How do elite freedivers achieve such remarkable breath-hold durations? The answer lies in a combination of physiological adaptations developed through years of training and specific preparation techniques employed immediately before record attempts. Enhanced lung capacity through cardiovascular training provides one foundation for extended breath holds. Competitive freedivers develop significantly larger lung volumes than average individuals, allowing them to begin their dives with greater oxygen reserves. Diaphragmatic breathing and respiratory muscle control further improve their efficiency. The technique of glossopharyngeal insufflation, also called lung packing, plays a crucial role in maximizing initial lung volume. This technique consists of forcing air into the lungs using glossopharyngeal muscle contractions similar to swallowing. Breath-hold divers perform this technique after a maximal inhalation prior to diving, thus increasing initial lung volume beyond what can be achieved through normal breathing. The process begins after filling the lungs to total lung capacity, then a mouthful of air with the glottis closed is compressed by the oropharyngeal muscles and then forced into the lungs. Before the pharyngeal muscles are relaxed, the glottis is closed to allow new air entering the oral cavity. This process can be repeated multiple times, packing additional air into the lungs with each cycle. Lung packing can increase lung volume by as much as 20 to 30% beyond normal total lung capacity, providing a substantial oxygen reserve for the dive. However, this technique carries risks, as forcing air into already full lungs creates significant pressure that could theoretically lead to lung rupture or other complications such as pneumomediastinum. Elite freedivers train extensively to safely perform this technique and have developed adaptations in lung flexibility that allow them to tolerate the increased pressure. Many deep freediving athletes focus extensively on improving lung flexibility as part of their training to prepare for the increased pressure at depth. At 80 meters deep, a diver experiences 9 bars of pressure as opposed to 1 bar at the surface, meaning 9 times more pressure is subjected to the lungs. Athletes use both wet exercises in water and dry exercises on land to increase lung compliance. Lung stretches on empty lungs and on full lungs are considered essential in training to adapt to depth. Exhale training for deep diving helps the lungs adapt by starting depth dives on either a passive exhale or full exhale, allowing the diver to expose themselves to the kinds of compressed lung volumes they would experience at target depths while diving to significantly shallower depths. Desensitization to high carbon dioxide and low oxygen levels represents another critical adaptation in elite freedivers. The urge to breathe is triggered primarily by rising carbon dioxide levels rather than falling oxygen levels. Through training, freedivers learn to tolerate much higher carbon dioxide concentrations and suppress the urge to breathe. This involves both physiological adaptation at the cellular level and psychological training to remain calm despite intense respiratory signals. The physiological break point, where the diaphragm begins involuntary contractions signaling the urgent need to breathe, can be pushed further through conditioning. Before world record attempts, freedivers employ pre-oxygenation techniques that involve breathing pure oxygen at 100% concentration for extended periods before the dive. This saturates the blood with oxygen far beyond what is possible breathing normal air, which contains only about 21% oxygen. The enhanced oxygen loading provides additional reserves that extend breath hold duration. However, this technique is strictly regulated in competitive freediving, with different rules applying to different record categories. Training-induced adaptations provide partial explanation for the greater breath hold performance seen in competitive freedivers compared with less trained freedivers and non-divers. Breath hold training alone may improve lung volumes and competitive performance. Forced expiratory volume in 1 second could be increased throughout a 3-month apnea training program, with concomitant increases in maximal oxygen uptake and arterial oxygen saturation during incremental exercise testing. Apnea training improves effectiveness at both peak exercise and submaximal exercise, and can also improve swimming technique by promoting greater propulsive continuity. Beyond trained athletes, certain human populations have developed genetic adaptations for diving over many generations. The Bajau people, often called sea nomads, of Southeast Asia spend their whole lives at sea, working 8-hour diving shifts with traditional equipment and short breaks to catch fish and shellfish. For hundreds or even thousands of years, the Bajau have lived at sea, and natural selection may have made them genetically stronger divers. These people regularly dive to depths of approximately 200 feet and can remain submerged for up to 13 minutes. Research has revealed that the Bajau possess enlarged spleens approximately 50% larger than those of the neighboring Saluan people, who do not traditionally dive. This difference exists even in Bajau individuals who do not actively dive, eliminating the possibility that larger spleens are simply a plastic response to diving activity. The genetic basis for this adaptation has been traced to a gene called PDE10A, present in the Bajau but absent or different in the Saluan. In studies involving mice, this gene has been associated with spleen size, as mice engineered to produce lower levels of the related thyroid hormone exhibited smaller spleens. The enlarged spleen provides a significant diving advantage because the spleen acts as a reservoir for red blood cells. During the diving response, the spleen contracts and releases its stored red blood cells into circulation, temporarily increasing the blood's oxygen-carrying capacity. The Bajau, with their 50% larger spleens, could mobilize perhaps 10% more red blood cells during a dive than those not adapted to the low oxygen conditions of breath-hold diving. This represents a clear example of recent human evolution in response to aquatic lifestyle demands. Another population showing genetic adaptations to diving is the Hinayeo, a group of all-female divers from the Korean island of Jeju. These women are renowned for their ability to dive in frigid waters without the aid of breathing equipment, even while pregnant. Studies show that the divers' remarkable abilities are due to both training and genetic adaptation, including gene variants associated with cold tolerance and decreased blood pressure. Genomic analysis revealed 2 gene variants in the Hinayeo that may help them cope with the pressures of diving, making the Hinayeo the second known population of traditional breath-hold divers that has evolved for diving. One gene is associated with cold tolerance, which could make the divers less vulnerable to hypothermia. The other gene is associated with decreased diastolic blood pressure, meaning blood pressure between heart contractions. The variant was found in 33% of participants from Jeju, but only 7% of mainland participants. The Hinayeo also showed pronounced bradycardia, or slowing of heart rate, when they dived, but this trait is likely due to a lifetime of training rather than genetics. Studies on Hinea, spanning several decades, have documented their thermoregulatory responses in terms of agging and cold adaptation. Older Hinea still retain local cold tolerance on the extremities despite their agging. Finger cold tests supported more superior local cold tolerance for older Hinea than for older non-diving females. During overall cold air exposure, more stable core temperature, lower mean skin temperature, and smaller energy metabolic rate of older Hinea indicate that older Hinea retain general cold adaptive traits. An interesting finding was the possibility of positive cross-adaptation, which might be supported by greater heat tolerance and cutaneous warm perception thresholds of older Hinea who adapted to cold water. Even human infants demonstrate innate aquatic responses that hint at our mammalian heritage. The diving response exists in human infants and includes an immediate bradycardic response, suggesting vagal mediation. Studies involving 36 infants during diving exercises in infant swimming showed that all participating infants displayed an immediate decrease in heart rate when submerged. On average, heart rate decreased by 25%, with a range from 5% to over 50%. The bradycardia was sustained during the dive and for some seconds afterwards, often followed by a tachycardia as the bradycardia ceased. A decline of reflex bradycardia was observed with increasing age, but the response was still clearly evident in infants over the age of 6 months, older than previously reported in earlier studies. This demonstrates that the diving response is not simply a neonatal reflex, but persists through infancy and indeed throughout life, though it may become less pronounced with age in the absence of training. The existence of this response in infants suggests it is a deeply programmed mammalian trait rather than a learned behavior. Human swimming ability itself represents an interesting baseline capability. While humans are not naturally gifted swimmers compared to aquatic or semi-aquatic mammals, we are considerably more competent in water than many terrestrial mammals. Our upright posture and mobile shoulders allow for effective arm stroking motions that generate propulsion. Studies comparing different swimming strokes have shown that front crawl is more efficient and has smaller active drag than backstroke. Stroke frequency in front crawl is approximately 3.5 to 7.7% lower than backstroke, while stroke length is approximately 5.9 to 11.9% longer. Froude efficiency, a measure of propulsive efficiency, is approximately 28.6 to 33.7% larger in front crawl than backstroke. Biomechanical analysis reveals that swimming performance depends on the balance of biomechanical and bioenergetic parameters. The main biophysical predictors of swimming velocity can be included in both biomechanical and physiological models, each explaining approximately 98% of variance. This suggests that both technique and physiology contribute substantially to swimming capability. Trained swimmers achieve significantly better efficiency than untrained individuals through improved technique and physiological adaptations. Current humans also possess voluntary breath control that surpasses most terrestrial mammals. While the extreme durations achieved by elite freedivers require extensive training, even average individuals can voluntarily hold their breath for periods that would be difficult or impossible for most land mammals. This voluntary control extends beyond simple breath holding to include the ability to precisely modulate breathing patterns, which is essential for both speech production and swimming coordination. Humans show some capacity for underwater vision, though limited compared to truly aquatic animals. The Moken children of Southeast Asia have demonstrated trainable underwater visual acuity through pupil constriction adaptation. Studies showed that European children could also learn to improve their underwater vision through training, developing similar pupil constriction responses. This suggests that human vision has some plasticity for aquatic adaptation, though our eyes fundamentally evolved for aerial vision and function suboptimally underwater without technological aids such as goggles or masks. The body fat distribution in humans, while not approaching the blubber of marine mammals, does provide some buoyancy that aids in swimming and floating. Humans can float relatively easily compared to many terrestrial mammals, partly due to our subcutaneous fat layer and partly due to air in our lungs. This buoyancy reduces the energy cost of staying at the surface and makes swimming less energetically demanding than it would be otherwise. These existing capabilities represent the starting point for any hypothetical aquatic evolution in humans. The mammalian diving reflex, trainable breath hold ability, genetic adaptations appearing in diving populations, infant diving responses, swimming competence, voluntary breath control, adaptable vision, and buoyancy from body fat all provide foundations that could be elaborated through evolutionary processes. The examples of the Bajau and Bajau demonstrate that human populations can evolve measurable physiological changes within relatively recent timeframes, on the order of hundreds to thousands of years, when strong selective pressures favor aquatic adaptations. However, these current capabilities should not be mistaken for true aquatic specialization. Humans remain fundamentally terrestrial mammals with only modest aquatic abilities. Our breath hold capacity, even in elite divers, measures in minutes rather than the hours achieved by deep-diving marine mammals. Our swimming efficiency is poor compared to seals or dolphins. We have no insulation adequate for extended time in cold water without artificial protection. We lack streamlined body shapes, propulsive tails, or flippers. Our sensory systems function poorly underwater. In short, current human aquatic capabilities represent potential starting points for evolution, but would require dramatic elaboration to achieve genuine aquatic specialization comparable to other marine mammals. Chapter 4: Respiratory System Transformation. The respiratory system presents perhaps the greatest challenge for any mammal attempting to transition from terrestrial to aquatic life. Air-breathing animals face a fundamental constraint when entering water. They must return to the surface to breathe. This tethering to the surface limits how deep they can dive, how long they can remain submerged, and ultimately determines their ability to fully exploit aquatic environments. The evolutionary transformation of the respiratory system in aquatic mammals represents one of the most dramatic and essential modifications required for marine life. Current human respiratory limitations underwater are severe. Without training, the average person can hold their breath for 30 to 90 seconds. Even elite freedivers, after years of training and using pre-oxygenation techniques, achieve breath holds measured in tens of minutes rather than hours. In contrast, some marine mammals can remain submerged for extraordinary durations. Sperm whales regularly dive for 60 to 90 minutes and have been recorded on dives lasting over 2 hours. Elephant seals routinely dive for 20 to 30 minutes and can extend dives to 2 hours when necessary. Beaked whales have been documented on dives exceeding 3 hours. How do marine mammals achieve such remarkable breath hold endurance? The answer lies in multiple coordinated adaptations affecting oxygen storage, oxygen consumption, and metabolic regulation. For humans to evolve similar capabilities, each of these systems would need to undergo substantial modification. Increased blood volume represents one crucial adaptation in diving mammals. Marine mammals possess significantly greater blood volumes relative to body size compared to terrestrial mammals. This expanded blood volume provides a larger reservoir for oxygen storage. Along with increased volume comes elevated hematocrit, the proportion of blood volume occupied by red blood cells, and elevated hemoglobin levels within those red blood cells. More red blood cells carrying more hemoglobin molecules means substantially greater oxygen-carrying capacity per unit of blood. The spleen plays a supporting role in this system by acting as a reservoir for concentrated red blood cells that can be released during dives. As we saw with the Bajau people, spleen size can evolve relatively quickly in response to diving demands. Marine mammals possess spleens that are proportionally larger than terrestrial mammals, and these spleens contract during dives to inject highly concentrated red blood cells into circulation, temporarily boosting oxygen-carrying capacity when it is needed most. Beyond blood, marine mammals store substantial oxygen in their muscles through elevated concentrations of myoglobin. Myoglobin is the protein responsible for binding and storing oxygen within muscle tissue, analogous to hemoglobin in blood. Diving mammals possess myoglobin concentrations that are dramatically higher than terrestrial mammals. Studies show considerable increases in myoglobin concentration within muscle cells, enhancing muscle oxygen storage and extending dive durations. The role of myoglobin in the evolution of mammalian diving capacity has been studied extensively. After the Devonian tetrapod land invasion, groups of terrestrial air-breathing and endothermic mammals repeatedly went back to live in the sea, relying on air intake at the surface for extended breath-hold dives to forage underwater, often at great depths and even in the coldest oceans. This adaptation has led to considerable increases in the concentration of myoglobin within muscle cells. Myoglobin content in cetaceans ranges from 1.81 to 5.78 grams per 100 grams of wet muscle. Myoglobin content and body mass are both positively and significantly correlated to maximum dive duration in toothed whales, though this differs from the relationship in baleen whales. The combined effects of body mass and myoglobin content account for approximately 50% of the variation in cetacean diving performance. Independent analysis of toothed whales shows that body mass and myoglobin content account for 83% of the variation in dive capacity. One physiological adaptation for prolonged dive duration in marine mammals is an elevated myoglobin concentration in skeletal muscle. Research modifying aerobic dive limit models for Weddell seals and running them for 4 different myoglobin concentrations, representing 7%, 50%, 100%, and 200% of normal Weddell seal skeletal muscle concentration, demonstrates the profound impact of myoglobin on diving capability. The evolution of myoglobin for deep-sea adaptation involved both common and unique strategies across different diving mammal lineages. Humans currently have modest myoglobin concentrations compared to marine mammals. For aquatic evolution to occur, myoglobin levels in human muscles would need to increase substantially. Some adaptation can occur through training. Studies on repeated scuba diving show that an adaptive response is triggered that affects cardiovascular, muscular, and immune biomarkers. However, the myoglobin increases achievable through training pale in comparison to the concentrations found in marine mammals, which require genetic changes affecting myoglobin production and regulation. Brain globins represent another adaptation for oxygen storage. Neuroglobin and cytoglobin, oxygen-binding proteins found in neural tissue, show elevation in diving mammals. These proteins help protect the brain from hypoxia during extended dives by providing local oxygen reserves within the brain itself. Physical activity, thermal demands, and hypoxia all contribute to increased production of these oxygen-binding proteins. Even with massively increased oxygen storage, marine mammals would quickly deplete their reserves without corresponding reductions in oxygen consumption. The solution involves profound metabolic rate reduction during dives. Deep-diving marine animals conserve oxygen through enhanced storage, profound metabolic rate reduction, selective blood flow, and adaptations for hypoxia tolerance. The dive reflex significantly reduces metabolic rate through bradycardia and peripheral vasoconstriction, prioritizing oxygen to vital organs. Initial studies involving restrained animals submerged forcibly led to a model suggesting a general decrease in aerobic and possibly overall metabolism to conserve limited oxygen stores for critical organs such as the nervous system and heart that are unable to endure severe hypoxia. This conservation is achieved through selective vasoconstriction, reducing blood flow to all body parts except oxygen-sensitive areas. In tissues receiving reduced blood flow, including skeletal muscles, anaerobic metabolism increases, resulting in lactic acid accumulation. This lactic acid is cleared into the bloodstream as the animal ventilates upon surfacing. Alongside selective vasoconstriction, heart rate reduces during submersion, leading to a corresponding decrease in cardiac output. This reduction typically matches the rise in peripheral resistance, maintaining stable central blood pressure. The cardiovascular changes during diving are substantial and coordinated. An extreme peripheral vasoconstriction is associated with a dramatic increase in arterial blood pressure, which in turn stimulates arterial baroreceptors, resulting in a substantial decrease in heart rate. Cardiac output is preferentially directed to essential organs, resulting in lactate accumulation in poorly perfused muscles. This selective perfusion ensures that limited oxygen supplies are directed where they are most critically needed. Marine mammals also tolerate hypoxia and lactic acid buildup far better than terrestrial mammals. They possess mechanisms to protect the brain from oxygen deprivation and can function with blood oxygen levels that would cause unconsciousness or death in humans. Cellular adaptations allow tissues to maintain function despite low oxygen availability, and metabolic pathways shift to favor anaerobic processes when necessary. For humans to evolve these respiratory and metabolic adaptations, substantial genetic changes would be required, affecting multiple systems simultaneously. Blood production mechanisms would need modification to increase red blood cell production and hemoglobin concentration. Spleen size and function would need enhancement. Myoglobin production in muscles would need dramatic upregulation. Cardiovascular control systems would require modification to produce more profound bradycardia and vasoconstriction. Cellular metabolism would need adjustment to tolerate hypoxia and manage lactate accumulation. These changes would occur gradually over many generations, with each small improvement in diving capability providing selective advantages. Beyond oxygen storage and metabolic reduction, the physical structure of the respiratory system itself would undergo transformation. One of the most dramatic changes in cetacean evolution was the migration of the nostrils from the front of the snout to the top of the head, forming the blowhole. This repositioning allows whales and dolphins to breathe while remaining almost completely submerged, minimizing the energy and time required for surface breathing. The evolutionary pathway for nostril migration is documented in the whale fossil record, showing gradual posterior movement over millions of years. The timeline for respiratory opening repositioning involves coordinated changes to skull anatomy. The premaxilla and other skull bones gradually reshape, with the nasal opening migrating dorsally and posteriorly with each successive species in the lineage. Pachycetus had nostrils in the typical terrestrial mammal position at the front of the snout. Ambulocetus showed slight posterior migration. Later species showed progressively more dorsal positions until reaching the fully posterior blowhole position in modern cetaceans. Anatomical skull modifications required for this transformation are extensive. The bones of the skull must be completely reorganized, with some expanding, others contracting, and the relationships between bones fundamentally altered. The nasal passages must be rerouted through the skull to connect the dorsally positioned blowhole with the lungs. This requires not just bone changes, but also modifications to surrounding soft tissues, including blood vessels, nerves, and muscles. Separated respiratory and digestive pathways represent another critical modification. In terrestrial mammals, including humans, the pharynx serves as a common passage for both food and air, with the epiglottis acting as a valve to prevent food from entering the airway. This arrangement makes choking possible and represents a significant design compromise. Marine mammals have evolved modifications that more completely separate these pathways, reducing the risk of water entering the lungs during feeding. Epiglottis modifications for underwater swallowing allow marine mammals to feed underwater without aspirating water. The larynx can form a tight seal with the nasopharynx, creating a continuous air passage from the blowhole to the lungs that bypasses the mouth entirely. This means that marine mammals can open their mouths underwater to capture prey without any risk of drowning. Larynx repositioning accompanies these changes. In humans, the larynx descends during development, creating the elongated vocal tract necessary for speech, but also creating the crossing of food and air passages. In marine mammals, the larynx remains in a more elevated position or is modified in ways that maintain separation between respiratory and digestive tracts during swallowing. Lung structure modifications for pressure resistance become essential for deep diving. The tremendous pressure at depth compresses the lungs, and without adaptations, this compression could cause injury. Marine mammal lungs are more compliant and flexible than terrestrial mammal lungs, allowing them to compress without damage. Reinforced alveoli provide structural support that prevents complete collapse while still allowing sufficient compression to avoid other problems. Partial lung collapse mechanisms at depth serve multiple functions. As a diver descends and the lungs compress, air moves from the alveoli into the larger airways and bronchi, which have rigid walls that resist collapse. This prevents nitrogen in the air from being absorbed into the bloodstream under high pressure, which is crucial for preventing decompression sickness. Marine mammals essentially avoid the bends by collapsing their lungs during deep dives, preventing problematic gas exchange at depth. Nitrogen absorption prevention represents a significant engineering challenge that marine mammals have solved through behavioral and anatomical adaptations. By exhaling before dives or allowing lung collapse at depth, they minimize nitrogen absorption that could form dangerous bubbles upon ascent. Their cardiovascular systems also possess features that help manage any nitrogen that does enter the bloodstream. For humans to evolve these respiratory system modifications, the timeline would extend over millions of years. The oxygen storage adaptations, including increased blood volume, elevated hemoglobin and myoglobin, and enlarged spleens, might appear relatively early, within the first several 100,000 years, as these can be achieved through relatively straightforward changes to existing systems. The Bajau example demonstrates that spleen size can evolve within thousands of years under strong selective pressure. Metabolic adjustments and enhanced diving reflexes would develop in parallel with oxygen storage improvements. The cardiovascular system would gradually evolve stronger vasoconstriction responses, more profound bradycardia, and better regulation of blood pressure during dives. Cellular adaptations for hypoxia tolerance would accumulate through changes affecting energy metabolism and oxygen sensing pathways. The physical restructuring of the respiratory tract, including nostril migration and pathway separation, would require much longer timeframes, likely millions of years. These changes involve coordinated modifications to multiple bones, extensive soft tissue reorganization, and developmental pathway alterations. The fossil record of whale evolution shows that these transformations occurred over approximately 8 to 10 million years, and similar durations would be expected for human aquatic evolution. Lung modifications for deep diving would develop gradually, with each improvement in compliance and pressure tolerance allowing slightly deeper or longer dives. The mechanisms for controlled lung collapse and nitrogen management would emerge through selection, favoring individuals who could dive deeper without experiencing decompression sickness or lung injury. Throughout this transformation, trade-offs would appear. Adaptations favoring aquatic respiration might compromise terrestrial respiratory function. Increased blood viscosity from elevated red blood cell counts could increase cardiovascular strain. Enhanced myoglobin concentrations in muscles might affect muscle function or metabolism. The evolutionary path would navigate these trade-offs, balancing aquatic advantages against any terrestrial disadvantages. With the balance shifting increasingly toward aquatic specialization as the lineage committed more fully to marine life. Chapter 5: Skeletal Restructuring for Aquatic Locomotion. The skeleton forms the structural framework upon which all other body systems depend. For terrestrial mammals, bones provide support against gravity, protect vital organs, anchor muscles for locomotion on solid ground, and store minerals essential for metabolism. When mammals transition to aquatic life, the selective pressures acting on the skeleton change dramatically. Gravity becomes nearly irrelevant as buoyancy supports body weight. Locomotion shifts from pushing against solid ground to undulating through three-dimensional fluid space. These fundamental changes drive profound skeletal restructuring that unfolds over millions of years. Terrestrial and aquatic locomotion impose completely different biomechanical demands. On land, limbs must bear body weight and generate forward thrust by pushing against the ground. The skeleton must be rigid enough to resist gravitational collapse while maintaining enough flexibility for efficient movement. In water, buoyancy eliminates weight-bearing requirements, but fluid resistance becomes the dominant challenge. Every movement through water encounters drag, the force that opposes motion through fluid. Minimizing drag while maximizing propulsive efficiency becomes paramount for aquatic animals. Streamlining represents the fundamental strategy for drag reduction in aquatic environments. A streamlined body optimizes pressure gradients that develop as an animal moves through water, allowing fluid to flow smoothly over the body surface rather than creating turbulent wakes that waste energy. Many fish species are dorsoventrally or laterally compressed, or long and torpedo-like, to minimize their impact against the fluid as they move. The shape of many fishes resembles modern airfoil profiles designed to minimize drag. This streamlined body form has evolved independently in multiple aquatic lineages, demonstrating convergent evolution toward optimal hydrodynamic shape. The two-dimensional horizontal cross-section through many fishes is similar in shape to modern airfoil profiles, and this shape represents an evolutionary optimization to minimize drag for economical swimming. Recent research has revealed that streamlined body shapes do not merely reduce drag passively, but can actually contribute directly to thrust generation. Airfoil-like mechanics generate thrust on the anterior body of swimming fish, meaning the front portion of a streamlined body actively pushes the animal forward rather than simply reducing resistance. This dual function of streamlining, both reducing drag and contributing to propulsion, helps explain why streamlined body forms have evolved so consistently across aquatic vertebrates. For humans to evolve aquatic locomotion capabilities approaching those of marine mammals, dramatic skeletal changes would be required. The vertebral column, which in humans is relatively stiff and specialized for upright posture, would need to become far more flexible to enable powerful swimming undulations. Cetacean evolution demonstrates this transformation clearly through the fossil record. The cetacean vertebral column underwent morphological modification for axial locomotion, representing one of many changes that occurred during and after the transition to aquatic life. Swimming in cetaceans is partially mediated by variations in vertebral morphology that creates trade-offs between body flexibility and rigidity. Forward propulsion of cetaceans is achieved by oscillation of the backbone, making the axial skeleton central to swimming performance. The vertebrae and spinal column of cetaceans underwent significant changes throughout their evolutionary history as they adapted to full-time aquatic lifestyle. Research on cetacean vertebral morphology reveals sophisticated adaptations for aquatic locomotion. Increased body flexibility in cetaceans is associated with increased maneuverability, which affects swimming speed and foraging efficiency. However, flexibility involves trade-offs. Some cetaceans have evolved rigid torsos for efficient fast swimming, while others maintain flexible torsos for enhanced maneuverability during hunting in complex environments. Deep-diving species show different vertebral adaptations compared to shallow divers, reflecting distinct ecological demands. Contrary to previous assumptions that cetacean backbones are homogeneous, research has revealed that the cetacean backbone is highly regionalized despite being uniform in shape along its length. Different vertebral regions show specialized structural properties suited to their specific functions. The thoracic region provides stability, the lumbar region generates propulsive power, and the caudal region forms the base for the tail flukes. This functional regionalization occurs through changes in vertebral morphology, intervertebral spacing, and bone microstructure. Studies of vertebral trabecular bone, the spongy bone tissue inside vertebrae, show that rigid torso shallow diving cetaceans have the greatest bone volume fraction and degree of anisotropy, while flexible torso deep diving species have the smallest values. These differences are driven by varying modes of caudal oscillation, swimming speed, and diving behavior. Despite having osteoporotic skeletons with lower bone density than terrestrial mammals, cetacean vertebral bone has greater microarchitectural variables compared to previously investigated terrestrial mammals. This reflects structural adaptation to support loads imparted on the vertebral column through caudal oscillatory swimming. Humans attempting to evolve aquatic locomotion would need vertebral modifications enabling powerful dorsoventral undulation. The current human spine, with its distinctive S-curve optimizing upright posture, would gradually straighten and lengthen. Individual vertebrae would become more uniform in shape, losing the regional specialization for bipedalism. Intervertebral discs would become more flexible, allowing greater range of motion. The attachment points for spinal muscles would enlarge dramatically to accommodate the massive muscles required for powerful swimming strokes. The evolution of cetacean tail flukes represents one of the most dramatic innovations in mammalian evolution. The most obvious adaptations in extant whales include a streamlined body, loss of hindlimbs, and development of a large muscular tail with a fluke that provides thrust. The bilaterally symmetrical flukes of cetaceans are supported by caudal tail vertebrae and dense connective tissues that are enveloped by skin. The cetacean tail flukes originated long after Lower Cambrian fin evolution and Devonian tetrapodal limb transition, with the first evidence for flukes appearing in the Eocene approximately 38 million years ago. The development of tail flukes likely arose from strong directional selection operating on a mutation governing the growth of a spur of connective tissue, driving it to expand and increase in rigidity at an evolutionarily rapid rate until it attained relatively modern form in basilosaurids and dorudontids. Within the fluke, collagen fibers are arranged in specific patterns that provide strength and flexibility. This attachment unites the caudal vertebrae associated with the flukes into a single resilient element. The enormous caudal muscles attached to the vertebrae via powerful tendons are responsible for the up and down movement of the fluke. For humans to develop tail flukes, the coccyx, our vestigial tail, would need to dramatically elongate through addition of numerous new vertebrae. These vertebrae would need to fuse into a rigid unit capable of supporting the massive stresses generated during swimming. Entirely new connective tissue structures would need to evolve laterally from these vertebrae, forming the horizontal flukes. Dense collagen fiber networks would develop within these flukes, arranged to optimize strength while maintaining appropriate flexibility. Powerful muscles would evolve along the entire lower spine and tail, dwarfing the muscles present in current humans. Limb transformation represents another critical aspect of aquatic skeletal evolution. The forelimbs of marine mammals have transformed into flippers through modifications of bone structure, proportions, and function. Recent research has identified adaptive evolution of BMP4 as a potential mechanism underlying cetacean flipper development. Vertebrate limbs have undergone profound morphological diversification, enabling adaptations to broad spectrums of ecological niches. In marine mammals, the evolution of highly specialized flipper-like forelimbs represents a profound structural transformation. The genetic basis for flipper evolution involves multiple developmental pathways. Cetacean hindlimbs were lost, and their forelimbs changed into flippers characterized by webbed digits and hyperphalangy, thus allowing them to live permanently in the ocean. The transition involves changes to genes controlling limb development, particularly those regulating interdigital tissue and bone growth. In terrestrial mammals, programmed cell death eliminates tissue between developing digits, creating separated fingers and toes. In aquatic mammals, this cell death program is suppressed or modified, allowing tissue to persist between digits and form webbing. Studies on human syndactyly, the condition where fingers or toes are webbed together, reveal the genetic mechanisms that could be co-opted for aquatic adaptation. Mutations in the homeodomain of HOXD13 cause syndactyly in humans. These mutations disrupt the normal developmental program that separates digits. The phenotype of second and third toe syndactyly maps to chromosome 2, demonstrating that relatively simple genetic changes can produce webbing between digits. In an aquatic evolutionary scenario, mutations that produce webbing would be favored by natural selection rather than being deleterious as they are in terrestrial humans. Beyond webbing, flipper evolution involves changes to bone structure within the limb. Hyperphalangy, the multiplication of finger bones beyond the normal number, occurs in cetacean flippers. While terrestrial mammals have a fixed number of phalanges per digit, typically 2 or 3, cetacean flippers can have 5, 7, or even more phalanges per digit. This multiplication of elements increases the length and flexibility of the flipper while maintaining structural support through the rigid bony skeleton. The forelimb bones themselves change proportion dramatically during aquatic evolution. The humerus, radius, and ulna become shorter and more robust, while the hand bones elongate. The entire limb rotates and flattens, transforming from a grasping appendage into a hydrodynamic paddle. Joints between bones become less mobile, with the elbow and wrist losing much of their range of motion. The flipper functions as a relatively rigid unit that can be rotated at the shoulder, but maintains a stable shape for efficient propulsion and steering. Hind limb reduction and eventual loss represents one of the most dramatic changes in cetacean evolution. The whale fossil record documents the gradual disappearance of hind limbs over millions of years. Pachycetus possessed 4 functional legs. Ambulocetus had hind limbs still capable of weight-bearing on land, but increasingly specialized for swimming. Later species showed progressively smaller hind limbs until they became non-functional externally and eventually disappeared entirely from the external body surface. Modern cetaceans retain vestigial pelvic bones embedded within their body wall. These bones no longer articulate with the spine and serve no locomotor function. Their persistence has been interpreted as evidence of evolutionary ancestry, demonstrating that whales descended from 4-legged terrestrial ancestors. Recent research has revealed that these vestigial pelvic bones may retain some function related to reproduction, serving as attachment points for muscles involved in mating. This demonstrates that even vestigial structures can be co-opted for new purposes. The genetic mechanisms underlying hind limb loss involve changes to developmental control genes. Evolutionary genetics of flipper forelimb and hind limb loss from genomes and transcriptomes in cetaceans reveals that limb development genes show signatures of relaxed selection or pseudogenization when their associated structures are lost. Genes essential for hind limb development in terrestrial mammals accumulate mutations that render them non-functional in cetaceans, as there is no longer selection to maintain their function. For humans evolving toward aquatic life, hind limb fate would depend on the locomotor strategy adopted. If tail-based propulsion became dominant, as in cetaceans, the legs might gradually reduce in size and eventually disappear. However, if a pinnape-like strategy persisted, with significant time still spent on land, the legs might transform into rear flippers while retaining substantial size and function. The evolutionary pathway taken would depend on ecological factors, including the degree of commitment to fully aquatic versus semi-aquatic lifestyle. Foot and hand webbing development would likely appear relatively early in the transition. Syndactyly represents a straightforward genetic change that could arise through mutations in developmental control genes. Fully webbed feet provide immediate swimming benefits, increasing the surface area of the foot and improving propulsive efficiency with each kick. Studies show that even partial webbing improves swimming performance measurably. Competitive swimmers with broader feet and hands naturally perform better. And full webbing would amplify this advantage substantially. Bone density changes accompany the transition to aquatic life. Terrestrial mammals require dense, mineralized bones to support body weight against gravity. Aquatic mammals, supported by buoyancy, can afford to reduce bone density. Correlation of bone density in aquatic and semi-aquatic animals to their lifestyle shows that fully aquatic mammals have lower bone density than terrestrial relatives. This reduction serves multiple functions, including reducing overall body weight, improving buoyancy control, and decreasing metabolic costs of bone maintenance. However, bone density changes are not uniform across the skeleton. Some bones, particularly those subject to high mechanical stresses like the vertebrae involved in swimming propulsion, maintain or even increase their density. The ribs of diving mammals show adaptations that allow them to collapse under pressure during deep dives, preventing lung damage. This requires specific changes to rib structure and the joints connecting ribs to vertebrae. The pelvis undergoes dramatic restructuring, narrowing substantially as the hindlimbs reduce, and the need for birth canal support changes with adaptations for aquatic birth. Ribcage expansion for increased lung capacity would occur gradually over many generations. Larger lungs provide greater oxygen storage for breath-hold diving, directly improving diving performance. The ribcage would expand both dorsoventrally and laterally, increasing total lung volume. The number of ribs might increase through duplication of vertebral segments, a change documented in some aquatic lineages. The sternum would elongate, and the connections between ribs and sternum would become more flexible, allowing greater expansion during inhalation. Spinal flexibility enhancement involves changes to intervertebral discs, the soft tissue pads between vertebrae. These discs would become thicker and more compliant, allowing greater bending of the spine during swimming undulations. The ligaments connecting vertebrae would strengthen to prevent injury during powerful swimming strokes while still permitting extensive flexion and extension. The overall length of the spine would increase through addition of vertebrae, particularly in the tail region where propulsive flukes develop. Skull reshaping for hydrodynamic efficiency would transform the human head from its current globular shape to a more streamlined profile. The forehead would slope more gradually, reducing drag as water flows over the head. The face would project forward less, creating a smoother profile. The nasal opening would begin its posterior migration, gradually moving from the front of the face toward the top of the head. This process would take millions of years, requiring coordinated changes to numerous skull bones. Eye socket repositioning might occur to optimize vision in the aquatic environment. In cetaceans, the eyes are positioned laterally, providing wide-angle vision useful for detecting predators and prey. The human forward-facing eyes, optimized for depth perception during tool use and manipulation, might shift laterally to improve peripheral vision underwater. Alternatively, they might remain forward-facing if tool use and complex manipulation remain important in the aquatic environment. Ear structure modifications for underwater hearing would transform the human auditory system substantially. Sound travels differently in water than in air, moving faster and over greater distances. The external ear, the pinna, would likely disappear as it serves no function underwater and creates drag. The ear canal would develop a mechanism for closing to prevent water entry. The middle ear bones and inner ear structures would modify to detect waterborne vibrations. In cetaceans, hearing involves bone conduction through specialized skull bones, and similar modifications might appear in aquatic humans. Melon organ, found in toothed whales, represents a specialized structure for echolocation. This fatty organ located in the forehead focuses sound waves for biological sonar. The morphology of the odontocete melon and its implications for acoustic function show that this structure evolved specifically for underwater sound production and focusing. If aquatic humans evolved echolocation capabilities, similar structures would need to develop through elaboration of fatty tissues in the forehead region. The timeline for complete skeletal restructuring would extend over millions of years, with different changes occurring at different rates. Some modifications, like webbing between digits or increased spinal flexibility, might appear within hundreds of thousands of years. Other changes, like complete hindlimb loss or full blowhole development, would require millions of years. The fossil record of whale evolution provides guidance for these timescales, showing that transformation from terrestrial quadruped to fully aquatic form took approximately 8 to 10 million years. Throughout this transformation, the skeleton would pass through intermediate stages similar to those documented in fossil whales. Early stages would show modest modifications compatible with both terrestrial and aquatic locomotion. Middle stages would display more pronounced aquatic specializations while retaining some terrestrial capability. Late stages would exhibit commitment to fully aquatic life with loss of terrestrial locomotor ability. The pace of change would depend on selection pressure intensity, with populations facing stronger aquatic selective pressures evolving more rapidly than those maintaining semi-aquatic lifestyles. Chapter 6: Skin, Hair, and Thermoregulation. The integument, comprising skin and its associated structures, forms the critical interface between an organism and its environment. For terrestrial mammals, skin provides protection from abrasion, prevents water loss, produces sensory structures including hair, and contributes to thermoregulation through fur insulation and evaporative cooling. When mammals transition to aquatic environments, the functions required of skin change dramatically. The integument must become waterproof to prevent excessive water absorption, must reduce drag for efficient swimming, must provide insulation in water rather than air, and must withstand constant immersion without degradation. These demands drive profound changes to skin structure and function. Body hair loss represents one of the most visible changes in aquatic mammal evolution. Humans already possess reduced body hair compared to other primates, a trait that has puzzled evolutionary biologists. Research comparing hair follicle density across primates reveals that hair density reduction occurred in the common ancestor of great apes, with further reduction specific to the human lineage. The reduced body hair that modern humans exhibit is unique among primates and represents a derived characteristic requiring explanation. Studies measuring hair follicle density show that humans have approximately the same number of hair follicles as chimpanzees, but human body hair is much finer and shorter. The density of hair follicles in hominids is lower than in Old World monkeys like macaques, suggesting that hair density reduction occurred after the split from Old World monkeys, but before the divergence of humans and chimpanzees. This means that partial hair reduction is ancestral to great apes, but the near-complete body hair loss in humans is a specifically human trait. Conversely, the density of eccrine glands in humans is, on average, 10 times greater than that in chimpanzees and macaques, both of which show remarkably similar densities. These results imply that a reduction in hair density occurred in the ancestors of humans and apes, followed by an increase in eccrine gland density and a decrease in fur coverage, specifically in humans. This evolutionary pattern demonstrates that hair loss and sweat gland proliferation occurred together in the human lineage, suggesting they are functionally linked adaptations for thermoregulation. For terrestrial humans, hair loss combined with increased sweating provides efficient evaporative cooling, advantageous for endurance activities in hot climates. However, for aquatic humans, this system would become obsolete. Sweating provides no cooling benefit in water, where evaporation cannot occur. The evolutionary pressure maintaining our current eccrine gland density would disappear, and these glands would likely reduce in number or disappear entirely, as they have in aquatic mammals. Aquatic mammals show dramatic hair loss through genetic changes affecting hair development. Genes lost during the cetacean transition from land to water include GSDMA, DSG4, DSC1, and TGM5, all involved in hair and skin development. These gene losses accompanied remodeling of epidermal morphology and the evolution of thick skin lacking sweat and sebaceous glands. The loss of genes previously considered essential demonstrates that evolutionary adaptation can proceed through gene deletion as readily as through gene modification or addition. The functional consequences of hair loss in water center on drag reduction. Hair creates substantial drag as water flows over the body surface, increasing the energy cost of swimming. Smooth, hairless skin allows water to flow more efficiently over the body, reducing drag and improving swimming performance. Studies of swimming efficiency in humans show that shaving body hair measurably improves performance, with competitive swimmers routinely removing body hair before major competitions. Complete genetic hair loss would provide even greater advantages. Enhanced physical barrier properties accompany hair loss in cetacean skin. Cetacean integument is uniquely suited for an aquatic lifestyle with epidermis that is smooth and rubber-like on the outside. On the inside, it is exceedingly thick and forms deep, root-like projections into the underlying dermis. This anatomy produces a dramatically increased basal to outer surface ratio and results in an expanded basal progenitor compartment, enabling a high epidermal turnover rate. The epidermal structure in marine versus freshwater cetaceans shows interesting variations. Two distinct strategies exist: lipid waterproofing versus thick and compact water-retaining approaches. Marine cetaceans in saltwater face osmotic pressure drawing water from their tissues, favoring lipid-based waterproofing. Freshwater cetaceans face osmotic pressure forcing water into their tissues, favoring thick, compact epidermis that physically resists water penetration. These different strategies demonstrate that aquatic skin evolution depends on the specific aquatic environment encountered. Rapid epidermal regeneration mechanisms allow cetacean skin to maintain integrity despite constant abrasion and biofouling challenges. Keratin expression for proliferation and repair shows upregulation in cetacean skin. The evolution of alternative splicing of certain keratins and differentiation-dependent upregulation of others have increased the complexity of keratin expression in cetacean epidermis. This reveals evolutionary diversification of the epidermal cytoskeleton and suggests a complete replacement of the quantitatively predominant epidermal proteins of terrestrial mammals by originally stress-inducible keratins in cetaceans. Gene loss in keratinization programs accompanies adaptation to aquatic environments. The cetacean epidermis maintains structural integrity through modified keratin expression patterns that differ fundamentally from terrestrial mammals. This represents not simply modification of existing programs, but wholesale replacement with alternative molecular mechanisms better suited to constant water immersion. Subcutaneous fat layer development represents perhaps the most critical thermoregulatory adaptation for aquatic life. Humans currently possess a modest layer of subcutaneous fat distributed relatively evenly across the body, unusual among primates. This continuous fat layer resembles the early stages of blubber development seen in semi-aquatic mammals. However, human subcutaneous fat averages only a few millimeters to perhaps 2 centimeters in thickness, far below the blubber thickness of marine mammals. Whale blubber is a thick layer of fat, specifically vascularized adipose tissue, that surrounds the body to keep vital organs warm in cold climates. Depending on the species, the thickness of blubber varies dramatically from 1 inch up to 11 inches thick. While the thickness of the blubber plays a role in determining what climates a marine mammal can comfortably inhabit, the blubber's lipid concentration is much more important for maintaining body heat than how thick the layer is. This means that both quantity and quality of fat matter for insulation. Blubber evolution in pinnipeds shows how this adaptation develops over evolutionary time. The evolutionary transition to blubber in pinnipeds involved development of a continuous subcutaneous adipose tissue layer reinforced with collagen and elastic fibers. This structural reinforcement prevents the blubber from simply redistributing under gravitational or hydrodynamic forces. The collagen network maintains blubber shape and position, ensuring consistent insulation across the body. The transition from fur to blubber as primary insulation represents a fundamental shift in thermoregulatory strategy. Fur insulates by trapping air close to the skin. This works well in air and can work in water if the fur remains dry. However, maintaining dry fur while swimming is energetically expensive and becomes increasingly difficult with extended submersion. Blubber insulates through the low thermal conductivity of fat itself, providing consistent insulation regardless of water immersion. This makes blubber far superior to fur for fully aquatic mammals. Insulation efficiency comparisons show that external insulation through fur becomes progressively less effective as aquatic commitment increases. Semi-aquatic mammals like otters maintain dense fur and spend significant energy grooming to maintain its insulating properties. Seals, more committed to aquatic life, use a combination of fur and blubber, with blubber becoming increasingly important in larger species. Fully aquatic cetaceans and sirenians rely entirely on blubber, having lost functional body hair completely. Thermoregulation in aquatic versus terrestrial environments faces fundamentally different challenges due to water's thermal properties. Water has thermal conductivity approximately 25 times greater than air, meaning heat flows from warm bodies into water much more rapidly than into air. This makes maintaining body temperature in water far more energetically costly than in air at the same temperature. The thermoneutral zone, the temperature range where an organism can maintain body temperature at resting metabolic rate without shivering or sweating, differs dramatically between air and water environments. For naked humans in air, the thermoneutral zone is 28 to 30 degrees Celsius. In water, it is considerably warmer at 34 to 35 degrees Celsius. This means that water temperatures below 35 degrees Celsius, which encompasses most of the world's oceans, cause heat loss in humans, requiring increased metabolic heat production. Five-fold faster heat loss in cold water compared to air creates serious challenges for aquatic mammals. For cold water immersions ranging from 18 degrees Celsius to 7 degrees Celsius. Body cooling rate has been shown to range on average between 0.5 degrees Celsius and 2.5 degrees Celsius per hour, respectively. Survival times are limited to 1 to 9 hours in lightly clothed individuals immersed in 5 degrees Celsius and 15 degrees Celsius. Metabolic changes during cold water immersion attempt to compensate for heat loss. Shivering thermogenesis versus exercise-based heat production represent two strategies for generating metabolic heat. Shivering generates heat through involuntary muscle contractions but impairs motor control and swimming ability. Exercise generates heat through voluntary muscle activity and maintains function but requires continuous effort. Free fatty acid increases during cold exposure provide fuel for thermogenesis. The body mobilizes fat stores to support increased metabolic demands of cold defense. Countercurrent heat exchange in flippers and flukes provides sophisticated thermoregulatory control in marine mammals. This system is shown to best effect in the flippers and tails of diving mammals, which must be thin and therefore free of insulating blubber if they are to function effectively for propulsion and steering. Approximately 1,000 arteries juxtaposed to 2,000 veins are found at the cranial end of the caudal vascular bundle in manatees, with this 1:2 ratio of arteries to veins persisting throughout the structure. The mechanism of countercurrent heat exchange involves transferring thermal energy from warm arterial blood flowing toward the periphery to cool venous blood returning from the periphery. Arterial blood loses heat to adjacent venous blood before reaching the flippers or flukes, while venous blood gains heat before returning to the body core. This traps heat in the body core and prevents excessive heat loss through uninsulated appendages. Arterial walls in these structures are relatively thin compared to typical arteries, facilitating heat. Marine mammals can modulate countercurrent heat exchange, allowing heat loss when necessary for cooling or conserving heat when necessary for warming. Activity in warm water creates metabolic heat that must be dissipated. The tail or flippers can transfer heat to the environment if arterial blood delivered to the skin is warmer than surrounding water. The ability to toggle this system on or off provides flexible thermoregulatory control suited to varying environmental temperatures and activity levels. For humans evolving aquatic adaptations, developing effective thermoregulation would be essential for survival. The subcutaneous fat layer would need to increase substantially, eventually reaching thicknesses measured in centimeters or even tens of centimeters, depending on the thermal environment. This blubber layer would need vascularization to provide metabolic support while maintaining low thermal conductivity. The collagen and elastic fiber networks reinforcing pinniped blubber would need to evolve, preventing the fat from redistributing inappropriately. Sweat and sebaceous glands would disappear as they serve no function in aquatic environments and potentially create vulnerabilities for infection or irritation. Gene losses affecting gland development would accumulate, eventually eliminating these structures entirely. The skin surface would become increasingly smooth and rubbery, optimized for minimal drag and rapid sloughing of biofouling organisms. Countercurrent heat exchange structures would develop in the limbs, particularly if they remain relatively thin for swimming efficiency. Networks of arteries and veins would become closely opposed, allowing heat transfer. The regulation of blood flow through these structures would become more sophisticated, providing fine control over heat loss or conservation. In cold water environments, this system would be essential for maintaining core temperature while allowing limbs to cool substantially. The epidermis itself would thicken dramatically, with deep projections into the dermis, creating a massively expanded basal cell layer. This would enable extremely rapid cell turnover, with the outer layers of skin being continuously replaced every few days or even hours. This rapid regeneration would combat biofouling and maintain the smooth surface essential for hydrodynamic efficiency. The molecular mechanisms of skin barrier formation would shift from the lipid bilayer system of terrestrial mammals to alternative mechanisms better suited to constant water immersion. The timeline for skin and thermoregulatory evolution would span hundreds of thousands to millions of years. Initial stages might show increased subcutaneous fat and reduced body hair within the first 100,000 years, as these changes provide immediate benefits and can occur through relatively simple genetic modifications. The Bajau and other diving populations demonstrate that some metabolic and physiological changes can occur within thousands of years under strong selection. Complete hair loss and sweat gland elimination would require longer timeframes, perhaps several 100,000 years, as multiple genes would need to be lost or inactivated. Thick blubber development, particularly reaching thicknesses comparable to marine mammals, would take even longer, requiring substantial changes to fat deposition patterns, connective tissue structure, and vascular supply. The most sophisticated adaptations, including countercurrent heat exchange systems and completely restructured epidermis with novel keratinization programs, would likely require millions of years to fully develop. These changes involve coordinated modifications to development, physiology, and anatomy that must all function together coherently. Chapter 7: Sensory System Adaptations. The sensory systems that animals use to perceive their environment are exquisitely tailored to the specific physical properties of the medium in which they live. Light, sound, chemical signals, and mechanical vibrations all behave fundamentally differently in water compared to air. For mammals transitioning from terrestrial to aquatic life, every sensory modality requires dramatic modification to remain functional in the new environment. Vision, hearing, smell, taste, and touch must all be restructured to detect and process information traveling through water rather than air. Some senses diminish or disappear entirely, while others become enhanced or develop completely new capabilities. Vision modifications for the underwater environment represent one of the most critical sensory adaptations. The fundamental challenge for underwater vision stems from the different refractive properties of water compared to air. In terrestrial mammals, including humans, the cornea provides the majority of the eye's focusing power. The curved surface of the cornea bends incoming light rays, focusing them through the lens onto the retina. This system works because of the large difference in refractive index between air and the corneal surface. Underwater, this corneal focusing power essentially disappears. Water and the aqueous humor filling the front chamber of the eye have nearly identical light-bending properties. When immersed in water, the small amount of bending that still occurs at the cornea's outer surface is almost totally reversed at its inner surface. This loss of corneal refractive power creates severe nearsightedness for animals whose eyes are adapted for aerial vision. Objects underwater appear blurry and out of focus because the eye cannot bend light rays sufficiently to focus them on the retina. Marine mammals have solved this problem through modifications to lens shape and size. The cetacean eye possesses a much more spherical lens compared to terrestrial mammals. This spherical shape provides greatly increased refractive power, compensating for the loss of corneal focusing. The lens is larger relative to eye size and positioned differently within the eye. These features characterize the dolphin eye as a basically aquatic one, rendering it right-sighted underwater. The cornea itself undergoes structural changes in aquatic mammals. Its outer surface has lower curvature than the inner one, meaning the cornea has the shape of a divergent lens. This unusual configuration further optimizes the optical system for underwater vision. The total refraction of the cornea and lens makes the cetacean eye well emmetropic, meaning properly focused for underwater vision. However, these same adaptations that optimize underwater vision create problems for seeing in air, making most marine mammals somewhat nearsighted when their heads emerge from water. Some aquatic animals have evolved the remarkable ability to accommodate or adjust focus for both underwater and aerial vision. Cormorants, birds that pursue fish underwater, have enlarged iris and ciliary muscles and very pliable lenses. The curvature of the front surface of the cormorant lens can increase by 64 diopters, which is more than required for overcoming the loss of corneal refraction. This extraordinary ability to accommodate vision allows cormorants to exploit both terrestrial and underwater habitats effectively. Dolphins have special muscles in the eye that are able to change the shape of the lens to adjust between seeing an object below water versus seeing an object in the air. The lenses and the cornea in a dolphin's eye naturally autocorrect for the difference in refraction of light underwater and above the surface. This dual adaptation provides these semi-aquatic mammals with functional vision in both environments, though neither is quite as sharp as the specialized single-environment vision of fully terrestrial or fully aquatic species. For humans evolving toward aquatic life, similar lens modifications would be essential. The currently flexible human lens, which changes shape to focus on objects at varying distances, would need to become more spherical and more powerful. The ciliary muscles controlling lens shape would strengthen and develop greater range of motion. If the evolving humans maintained any connection to terrestrial environments, accommodation capabilities similar to dolphins or cormorants would be advantageous, allowing functional vision both underwater and in air. Eye size changes represent another adaptation documented in vertebrate evolution between aquatic and terrestrial environments. The photoreceptor circuits of the eye underwent substantial reorganization during the evolutionary transition from water to land in early tetrapods. A reverse transition from land to water would require complementary changes. The distribution of photoreceptors within the retina would reorganize to optimize detection of available light underwater. The wavelength sensitivity of photopigments might shift, as water selectively filters different wavelengths of light compared to air. Protective adaptations for the eye become crucial in the marine environment. The nictitating membrane, often called the third eyelid, provides such protection in many aquatic animals. This translucent or transparent membrane sweeps horizontally across the eye, protecting the cornea from mechanical injury, debris, and irritation while still allowing vision. Aquatic animals require clear vision despite murky waters filled with particles, and penguins' transparent membranes enable them to hunt fish underwater without compromising eyesight. Sea turtles use theirs during swimming to guard against saltwater irritation. The nictitating membrane serves multiple functions, including protection from environmental aggression and providing lubrication to the cornea by distributing the precorneal film. For aquatic humans, developing such a structure would provide significant advantages, protecting the eyes from saltwater, suspended particles, and potential injury while diving. The membrane would need to be transparent to avoid interfering with vision, and would need to move quickly and frequently to keep the eye surface clean and moist. Tear composition would necessarily change for aquatic adaptation. Current human tears are hypotonic compared to seawater, designed to keep the eye surface moist in air. Aquatic mammals have modified tear composition that provides appropriate osmotic balance when eyes are constantly bathed in seawater. The lacrimal glands producing tears would need to modify their secretions, and the mechanisms for draining tears would likely reduce or disappear, as continuous flushing is less necessary when immersed in water. Hearing adaptations for underwater sound transmission represent another critical sensory transformation. Sound travels approximately 4 times faster in water than in air, and the mechanisms for detecting sound must be completely restructured for aquatic hearing. The most drastic changes in auditory systems between terrestrial and marine mammals can be found in the cetaceans. The whale ear, initially designed for hearing in air, became adapted for hearing underwater in less than 10 million years of evolution. This study describes the evolution of underwater hearing in cetaceans, focusing on changes in sound transmission mechanisms. The outer ear pinna and external auditory meatus were functionally replaced by the mandible and the mandibular fat pad, which posteriorly contacts the tympanic plate, the lateral wall of the bulla. In odontocetes, the lower jaw is surrounded by fats that, along with a thin bony area called the pan bone, are thought to conduct sound to the middle ear. Unlike land mammals that have ears attached to the skull, the middle and inner ears of cetaceans are encased in bones that are located in a cavity outside the skull. In odontocetes, these bones are attached to the skull by ligaments rather than solid bone connections. This isolation prevents sound from traveling through the skull bones, ensuring that sounds reach each ear independently, which is crucial for directional hearing. Changes in the ear include thickening of the tympanic bulla medially, isolation of the tympanoperiotic complex by means of air sinuses, functional replacement of the tympanic membrane by a bony plate, and changes in ossicle shapes and orientation. Pachystids, the earliest archaeocetes, had a land mammal ear for hearing in air and used bone conduction underwater, aided by the heavy tympanic bulla. Remingtonostids and protocetids were the first to display a genuine underwater ear, where sound reached the inner ear through the mandibular fat pad, the tympanic plate, and the middle ear ossicles. The basilar membrane within the cochlea shows remarkable adaptations related to frequency range. The thickness and width of cetacean basilar membranes are closely linked to the unique hearing capacities of toothed and baleen whales. The thicker and stiffer the basilar membrane, the more tuned an ear will be for higher frequency hearing. Toothed whales have evolved additional adaptations that increase the stiffness of the basilar membrane, including bony supports present in toothed whale cochlea. The thickness of the membrane is also larger compared to terrestrial mammals of the same body size. These adaptations contribute to the exceptionally high hearing range in toothed whales, which can detect ultrasonic frequencies far beyond human hearing capabilities. Baleen whales, on the other hand, have exceptionally broad, thin, and elastic basilar membranes. These characteristics contribute to the low-frequency hearing range in baleen whales, which can detect infrasonic frequencies below the range of human hearing. This allows communication over vast distances, as low-frequency sounds travel farther through water than high frequencies. For humans evolving aquatic hearing, the external ear would likely disappear first, as it serves no function underwater and creates drag. The ear canal would develop mechanisms for closing to prevent water entry during dives. The tympanic membrane might be replaced or supplemented by bone conduction pathways. Fat pads might develop in the jaw to channel sound to the inner ear, similar to cetaceans. The middle ear ossicles would modify their shapes and connections to efficiently transmit waterborne vibrations. The inner ear structures would adapt to the different frequency ranges most useful in aquatic environments. Echolocation development represents one of the most sophisticated sensory adaptations in marine mammals. Dolphins use echolocation or biosonar to navigate, locate food, and investigate their surroundings. They emit a series of clicks through their melon, their round forehead. The sound waves bounce off of objects and return to the dolphin, who receives the sound waves through their lower jawbone. The melon consists of fatty tissue and fluid and serves as the lens through which sound is focused during echolocation. Echolocation is seeing with sound, much like sonar on a submarine. They can only echolocate in the water and not through the air. Sound waves are created in the nasal sacs and focused through the melon at various frequencies, allowing the dolphin to see with sound. The morphology of the odontocete melon and its implications for acoustic function reveal a highly specialized structure. The melon is composed of specialized fats with specific acoustic properties that allow precise focusing of sound beams. Different species have melons of varying sizes and shapes related to their specific echolocation needs. Species hunting fast-moving fish in open water have different melon structures compared to species navigating complex reef environments or murky river waters. For humans to evolve echolocation capabilities, substantial new structures would need to develop. The ability to produce rapid, precisely timed clicks would require modifications to the nasal passages and development of specialized sound-producing structures. A melon-like organ would need to evolve in the forehead to focus these sounds. The auditory system would need to become capable of processing the returning echoes and extracting information about object size, distance, texture, and movement. This would require not just anatomical changes, but extensive neural modifications to process echolocation information. Olfaction reduction or loss is characteristic of aquatic mammals. Marine mammals have experienced large numbers of olfactory receptor gene losses compared with their related terrestrial lineages. Convergent degeneration of olfactory receptor gene repertoires in marine mammals results from the frequent birth and death evolution under varied functional constraints. All marine mammals had fewer olfactory receptor genes than their related terrestrial lineages, with the fewest olfactory receptor genes found in cetaceans, which may be closely related to olfactory degradation. Cetaceans lost a large amount of olfactory receptor genes when they became fully adapted to the water environment, because they are largely independent of the terrestrial environment throughout their life cycle. Pinnipedia and Sirenia, aquatic mammals, still retain large numbers of olfactory receptor genes because they have a certain dependence on terrestrial or shallow water environments in terms of reproduction or other important functions. The convergent loss of olfactory genes in marine mammals is most likely due to the unique olfactory system of aquatic mammals, which subjects most olfactory receptor genes to relaxed or no selective constraints. Smell, as terrestrial animals understand it, is nearly useless underwater. Airborne odor molecules cannot be detected while submerged. Cetaceans, particularly toothed whales, have reduced or absent olfactory bulbs. Some species have lost the olfactory structures entirely. This represents one of the most dramatic sensory losses in marine mammal evolution, but it comes with minimal cost since olfaction provides little benefit in the aquatic environment. Taste adaptation shows interesting patterns in marine mammals. Chemoreception in dolphins reveals that while traditional olfaction is lost, gustatory capabilities may persist or even expand. The authors of behavioral studies noticed that dolphins were able to perceive orally what other mammals perceive by smell. This suggests a shift from distance chemoreception through smell to contact chemoreception through taste. Massive losses of taste receptor genes in toothed and baleen whales show that taste has also been substantially reduced. Taste receptor genes are functionally important in animals, with a surprising exception in marine mammals, which show extensive losses of sweet, umami, and bitter taste receptor genes. Examining 3 genes responsible for taste signal transduction found the relaxation of functional constraints on taste signaling pathways along the ancestral branch leading to whales. Together, these results strongly suggest extensive losses of sweet, umami, bitter, and sour tastes in whales, and the relaxation of taste function most likely arose in the common ancestor of whales between 36 and 53 million years ago. Whales represent the first animal group to lack 4 of 5 primary tastes, probably driven by the marine environment with high concentration of sodium, the feeding behavior of swallowing prey whole, and the dietary switch from plants to meat in the whale ancestor. Interestingly, in contrast to the widespread pseudogenization of other taste receptor genes, researchers were able to identify 3 salty taste receptor genes with nearly complete coding regions and intact open reading frames in cetaceans. The genes have experienced strong purifying selection, and salty taste may be functional in whales. This makes sense given that marine mammals live in an environment with extremely high salt concentrations and would benefit from the ability to detect variations in salinity. For aquatic humans, olfaction would likely diminish substantially, with olfactory receptor genes accumulating inactivating mutations over generations. The olfactory bulbs in the brain would shrink. The nasal passages, being redirected for breathing through a dorsal blowhole, would lose their olfactory epithelium. Taste would also simplify, potentially retaining only salt detection while losing other taste modalities that provide less useful information when swallowing prey whole underwater. Chapter 8: Metabolic and Cardiovascular Changes. The metabolic and cardiovascular systems form the engine that powers all biological activity. These systems must deliver oxygen and nutrients to tissues, remove waste products, generate heat to maintain body temperature, and provide energy for movement and other functions. For aquatic mammals, the demands placed on these systems differ dramatically from those faced by terrestrial mammals. Extended breath-hold diving creates periods of limited oxygen availability, requiring sophisticated oxygen management. Swimming provides efficient locomotion, but requires different muscle types and energy systems. Temperature regulation in water demands far more metabolic energy than in air. These challenges drive profound modifications to metabolism and circulation. Basal metabolic rate adjustments for aquatic life involve fundamental changes to how the body budgets energy. Metabolic rate, the rate at which an organism consumes energy, determines how quickly fuel reserves are depleted and how much food must be consumed. Marine mammals show complex patterns of metabolic adaptation that vary with species, size, and energy expenditure in swimming versus walking or running differs substantially. Swimming can be energetically efficient for streamlined aquatic mammals, but is relatively costly for semi-aquatic animals that maintain features adapted for terrestrial life. The biomechanics and energetics of aquatic versus semi-aquatic mammals show clear distinctions. Wave drag, experienced by semi-aquatic surface swimmers, substantially increases energy costs compared to fully submerged swimming. Animals swimming at the surface must push through both water and the water-air interface, creating waves that carry away energy. Oxygen consumption differences between surface swimming and submerged swimming reflect these energetic costs. Surface swimming typically requires higher oxygen consumption per unit distance traveled compared to fully submerged swimming, especially at higher speeds. However, submerged swimming requires breath holding, creating a different set of constraints. The trade-offs between these strategies shape the diving patterns and foraging behaviors of marine. For humans evolving aquatic capabilities, metabolic efficiency during swimming would gradually improve through multiple mechanisms. Body shape would streamline, reducing drag. Muscle composition would shift toward types optimized for sustained swimming. Energy storage patterns would change to support aquatic activities. These changes would occur incrementally over many generations, with each small improvement in swimming efficiency providing selective advantages. Cardiovascular adaptations for diving build upon the mammalian diving reflex discussed earlier, but extend far beyond those basic responses. The cardiovascular system must manage profound changes in blood distribution, blood oxygen content, blood pressure, and heart function during dives. Heart structure modifications in diving mammals include enlargement of the heart relative to body size, increased muscle mass in the ventricular walls, and modifications to the cardiac conduction system that controls heart rhythm. Blood pressure regulation during dives presents significant challenges. Vascular responses to simulated breath hold diving show complex patterns. Peripheral vasoconstriction dramatically increases blood pressure as the same cardiac output is forced through a much smaller portion of the circulatory system. An extreme peripheral vasoconstriction is associated with a dramatic increase in arterial blood pressure, which in turn stimulates arterial baroreceptors, resulting in a substantial decrease in heart rate. This bradycardia helps prevent blood pressure from rising to dangerous levels despite the intense vasoconstriction. The balance between bradycardia and vasoconstriction maintains relatively stable central blood pressure while dramatically altering peripheral circulation. Cardiac output is preferentially directed to essential organs, resulting in lactate accumulation in poorly perfused muscles. The cardiovascular system must be capable of sustaining these dramatic changes for extended periods during long dives. Spleen contraction for red blood cell release provides an additional oxygen reserve. The spleen acts as a reservoir for concentrated red blood cells that can be injected into circulation when needed. During the diving response, the spleen contracts and releases its stored red blood cells, temporarily increasing the blood's oxygen-carrying capacity. The Bajau, with their 50% larger spleens, could mobilize perhaps 10% more red blood cells during a dive than those not adapted to the low oxygen conditions of breath-hold diving. Enhanced oxygen-carrying capacity through elevated red blood cell counts and hemoglobin concentrations provides greater oxygen storage in the blood. Increased blood volume gives marine mammals larger oxygen reserves. Together with elevated hematocrit and hemoglobin levels, this substantially increases total oxygen storage. The blood essentially functions as an oxygen tank that can supply tissues during breath holds. Anaerobic metabolism tolerance becomes crucial during extended dives. When tissues are deprived of oxygen, they shift to anaerobic metabolic pathways that produce energy without consuming oxygen. The main byproduct of anaerobic metabolism is lactic acid, which accumulates in tissues and must eventually be cleared. Marine mammals tolerate much higher lactate levels than terrestrial mammals. Their tissues can function despite acidic conditions that would impair or damage terrestrial mammal tissues. Lactic acid buildup management involves several strategies. During dives, lactate accumulates primarily in skeletal muscles that are deprived of blood flow through vasoconstriction. This lactate remains sequestered in the muscles until the animal surfaces and normal circulation resumes. Upon surfacing, the accumulated lactate is gradually cleared into the bloodstream as the animal ventilates. The liver converts lactate back to glucose through gluconeogenesis, recycling the byproduct of anaerobic metabolism into useful fuel. Blood chemistry changes for diving include modifications to buffering capacity, allowing marine mammals to tolerate greater acidosis. pH regulation during extended breath holds requires enhanced buffering systems in the blood and tissues. Marine mammals possess higher concentrations of bicarbonate and other buffers that neutralize the acids produced during anaerobic metabolism. Carbon dioxide tolerance mechanisms allow marine mammals to withstand much higher carbon dioxide levels than terrestrial mammals. In terrestrial mammals, rising carbon dioxide triggers an overwhelming urge to breathe, usually long before oxygen depletion becomes dangerous. Marine mammals have modified chemoreceptors that are less sensitive to carbon dioxide, allowing them to tolerate elevated levels without distress. This shifts the limiting factor for dive duration from carbon dioxide accumulation to oxygen depletion. Nitrogen handling and decompression present challenges for deep-diving mammals. When air-breathing animals dive, nitrogen from the lungs dissolves into the bloodstream under pressure. If the animal ascends too quickly, this dissolved nitrogen can form bubbles in the blood and tissues, causing decompression sickness or the bends. Marine mammals avoid this problem through several mechanisms, including exhaling before diving, lung collapse at depth that prevents gas exchange, and possibly tissue modifications that reduce nitrogen solubility or enhance bubble clearance. Muscle composition shifts dramatically in aquatic mammals. Slow-twitch versus fast-twitch fiber ratios change to favor sustained activity. Slow-twitch fibers use oxygen efficiently and resist fatigue, but generate less power than fast-twitch fibers. Fast-twitch fibers produce powerful contractions, but fatigue quickly and rely more heavily on anaerobic metabolism. Marine mammals adapted for sustained swimming have high proportions of slow-twitch fibers, while those that rely on burst swimming for prey capture maintain more fast-twitch fibers. Myoglobin density in swimming muscles reaches extraordinary levels in diving mammals. Myoglobin content in cetaceans ranges from 1.81 to 5.78 grams per 100 grams of wet muscle. This is several times higher than terrestrial mammals. Myoglobin content and body mass are both positively and significantly correlated to maximum dive duration in toothed whales. The combined effects of body mass and myoglobin content account for approximately 50% of the variation in cetacean diving performance. The evolution of myoglobin for deep-sea adaptation involved both common and unique strategies across different diving mammal lineages. After the Devonian tetrapod land invasion, groups of terrestrial air-breathing and endothermic mammals repeatedly went back to live in the sea, relying on air intake at the surface for extended breath-hold dives to forage underwater, often at great depths, and even in the coldest oceans. This adaptation has led to considerable increases in the concentration of myoglobin within muscle cells. Efficient propulsion, muscle development, creates the powerful musculature required for swimming. The muscles driving the tail flukes in cetaceans or the flippers in pinnipeds are among the most massive in the body. These muscles must generate tremendous force repeatedly throughout each dive, propelling streamlined bodies through resistant water. The attachment sites on bones must be reinforced to withstand these forces, leading to the enlarged muscle attachment processes seen in marine mammal skeletons. Energy storage strategies in marine mammals differ from terrestrial mammals. Fat deposition patterns shift to create the subcutaneous blubber layer that provides both insulation and energy storage. Unlike terrestrial mammals that might store fat in discrete deposits, such as around internal organs or in specialized adipose pads, marine mammals distribute blubber continuously across the body surface. This serves the dual purposes of thermoregulation and fuel. Glycogen storage modifications allow muscles to maintain larger reserves of immediately available energy. Glycogen, the storage form of glucose, provides quick energy during intense activity. Marine mammals have enhanced glycogen storage in muscles and liver, supporting both burst swimming for prey capture and sustained swimming during migrations. Fasting adaptations for long migrations are particularly well developed in baleen whales and elephant seals. These animals undergo extended fasts lasting weeks or months while migrating, breeding, or molting. During fasts, they rely entirely on stored energy, primarily blubber. Their metabolic systems are adapted to efficiently mobilize these fat stores while protecting vital protein reserves. Ketone bodies, produced from fat metabolism, become the primary fuel for many tissues, including the brain. The metabolic rate may actually decrease during fasting, conserving energy reserves. Hormonal changes suppress appetite and alter metabolism to favor fat utilization over protein catabolism. These adaptations allow marine mammals to survive extended periods without food while remaining active and maintaining body condition. For humans evolving toward aquatic life, metabolic and cardiovascular changes would accumulate gradually. Early changes might include increased blood volume, elevated red blood cell production, enlarged spleen, and enhanced diving reflex. These could appear within thousands to tens of thousands of years, as demonstrated by the Bajau adaptations. Increased myoglobin concentrations in muscles would develop over longer timeframes, perhaps hundreds of thousands of years, requiring changes to genes regulating myoglobin production. More profound changes, such as dramatically altered heart structure, sophisticated vasoconstriction control, extreme lactate tolerance, and complete reorganization of muscle fiber types, would require longer evolutionary periods measured in hundreds of thousands to millions of years. The coordination required between cardiovascular, respiratory, and metabolic systems means these changes must occur in concert, with each system's modifications complementing those of the others. The timeline would depend heavily on selection pressure intensity. Populations facing immediate survival challenges related to aquatic foraging might evolve more rapidly than those maintaining mixed terrestrial aquatic lifestyles. Genetic variation within the population would also influence evolutionary rate, as adaptive changes can only occur if the necessary genetic variants arise through mutation and are available for selection to act upon. Chapter 9. Reproductive and developmental adaptations. Reproduction and development in aquatic environments present unique challenges that terrestrial mammals never face. The fundamental question of how to give birth to air-breathing offspring underwater has been solved independently by multiple marine mammal lineages, each developing sophisticated adaptations. The newborn must avoid drowning during delivery, reach the surface immediately for its first breath, maintain body temperature in water, and develop swimming abilities rapidly. Maternal care must accommodate an aquatic environment where traditional terrestrial nursing positions are impossible. These challenges have driven remarkable evolutionary innovations that would be required for any human population transitioning to marine life. Aquatic birth mechanisms in cetaceans represent some of the most dramatic adaptations for marine reproduction. The position of the fetus during delivery is crucial for survival. In whales and dolphins, the calf typically emerges tail first, a presentation unique to cetaceans. This fluke-first position helps prevent the calf from drowning during the underwater birth. The reason the cetacean is always born tail first is due to distribution of weight in the fetus. Since the tail is the lighter part of the fetus, it will rise to a higher point than the head and body. Due to the layout of the uterus, the highest point leads towards the cervix. In most terrestrial mammals, the opposite is true. The hindquarters tend to be heavier, and the head will point towards the opening of the cervix. The mother whale positions herself vertically for the birth, allowing the calf to rise to the surface more easily. This vertical positioning is not accidental, but represents an evolved behavior that facilitates safe delivery in the aquatic environment. Once the calf is born, it must quickly adapt to its aquatic environment, with the first moments after birth being critical for its survival. Recent observations have captured humpback whale births on film, revealing details of this remarkable process. In one documented case, a tiny tail was observed sticking out of the female's genital slit during labor. Tail-first delivery is a critical adaptation that ensures the calf's blowhole, the nostril, is the last part to emerge, minimizing the risk of drowning during the birthing process. If the head emerged first, as in most terrestrial mammal births, the calf could attempt to breathe while still partially inside the birth canal, aspirating water and drowning. By emerging tail first, the entire body can be delivered before the head and blowhole exit, ensuring the calf takes its first breath only after being completely born. Immediately after birth, the mother dolphin, often with assistance from other females in the pod known as aunties, quickly guides or pushes the newborn calf to the surface to take its crucial first breath. In some cases, if the mother is struggling with the birth, the auntie will help by pulling the calf out, since they are often born tail first. This cooperative assistance demonstrates the social complexity of marine mammal reproduction and the importance of experienced individuals in ensuring neonatal survival. Umbilical cord breaking mechanisms in cetaceans differ from terrestrial mammals. The cord is extremely tough, like thick sinew, requiring significant force to sever. In marine mammals, the umbilical cord typically breaks naturally during or immediately after birth, either through the force of delivery or through the mother's movements as she assists the calf to the surface. In harbor seals, researchers have documented pups with fresh bleeding umbilical cords at age 0, with the condition of the umbilical cord used to determine age up to 3 days. During a uterine contraction, flow through the umbilical cord is maintained. Wharton's jelly, the gelatinous substance surrounding the blood vessels in the cord, acts as an oxygen reserve, which delays the onset of anaerobic metabolism and ultimately delays the onset of fetal acidosis. This buffering capacity is particularly important during aquatic births, where delivery may take longer due to the mechanical challenges of underwater parturition. Birth canal adaptations for underwater delivery involve modifications to the female reproductive tract. The uterine contractions and placental perfusion reduction during labor mirror the dive response in human fetuses, suggesting pre-adaptations that could be elaborated for fully aquatic birth. The muscular contractions that expel the fetus must be powerful enough to deliver a large calf through water resistance, while the mother maintains her own position and breathing. Calf orientation during the final stages of delivery is precisely controlled. The tail-first presentation ensures that as the widest part of the calf, the head, passes through the birth canal, the entire delivery accelerates to completion. This prevents the calf from becoming stuck partway through delivery, a potentially fatal complication in the aquatic environment where the partially delivered calf could drown. Maternal assistance in the first breath is essential. The mother, and often other pod members, actively guide the newborn to the surface immediately after birth. The calf must learn instantly to use its blowhole for breathing rather than attempting to breathe through its mouth. This behavioral adaptation occurs through a combination of instinct and immediate learning, facilitated by the mother's guidance. Placental structure in marine mammals shows specialized features suited to the aquatic environment. The placenta must supply oxygen and nutrients to the developing fetus throughout pregnancy, which in cetaceans can last 10 to 18 months, depending on species. Steroidogenic enzyme localization in whale placentas shows morphological changes through gestational stages. The placenta of marine mammals must support extended gestation periods that produce highly precocial offspring capable of swimming and breathing independently from birth. Placental expulsion occurs after the calf is born and has reached the surface to breathe. Floating placentas are evidence of recent births, with humpback whale placentas documented as giant blobs floating in the ocean. The placenta may measure several feet across and weigh dozens of kilograms, representing a substantial mass of tissue that must be expelled after delivery. Gestation period lengths in cetaceans vary by species, but are generally long compared to terrestrial mammals of similar size. Recent research indicates that bottlenose dolphin pregnancy lasts about 12 months. Among whales, minke whales have relatively short gestation at 10 months, while killer whales have the longest at 15 to 18 months, with an average of about 17 months. Sperm whales carry their young for 14 to 16 months. Blue whales and humpback whales both have gestation periods of 11 to 12 months. Beluga whales and narwhals both have 14 to 15-month gestations. Gray whales carry their calves for approximately 13.5 months. These extended gestation periods allow for substantial fetal development, producing offspring that are large, well-developed, and capable of swimming from birth. This precocial development is essential for survival in the aquatic environment where helpless newborns would quickly drown or succumb to hypothermia. Lactation modifications for aquatic nursing represent another critical reproductive adaptation. Marine mammals produce high-fat, energy-rich milk that facilitates rapid growth of offspring. Maternal provisioning in pinnipeds involves milk with dramatically different composition than terrestrial mammals. Water content of pinniped milk ranges from approximately 35 to 65% compared with cow and human milk that contains approximately greater than 85% water. In contrast, fat content of pinniped milk ranges from 25 to 60% lipid compared with the 4% milk fat in cow or human milk. In marine and aquatic carnivores, including marine mammals, this results in milks containing very large quantities of nutritionally significant lipids. Milk lipids range from 0.2% in the black rhinoceros to 60% in the milks of some faucid seals. In all species studied, milk lipid is composed primarily of triacylglycerols. The fatty acid composition of marine mammal milk includes essential fatty acids that can only arise from maternal diet and must be present in any artificial formula. Whether the more abundant longer-chain omega-3 and omega-6 polyunsaturated fatty acids are essential to neonatal marine mammals is unclear, but given that their natural milks are absolutely saturated with these compounds, they presumably convey some advantage. Sea lions produce milk that is generally somewhat less concentrated, containing 41 to 63% dry matter, 32 to 50% fat, and 8 to 10% protein. Milks of the phocids, the true seals, contain 66 to 71% dry matter, 47 to 61% fat, and 5 to 10% protein. Large changes occur from early to mid-lactation in many species, with milk composition shifting to meet the changing needs of growing offspring. Forceful milk ejection underwater allows marine mammals to nurse despite water immersion. The mammary glands have specialized muscles that forcefully eject milk directly into the calf's mouth, preventing dilution by seawater. The calf forms a seal around the nipple with its tongue and mouth, and milk is essentially squirted into the calf's throat. This prevents the calf from needing to create suction, which would be difficult underwater and might result in water aspiration. Nursing behavior in water requires specific positioning and coordination between mother and calf. The calf must locate the mammary glands, which are positioned in mammary slits on the mother's ventral surface near the genital opening. The calf swims close to the mother, often upside down, to access the nipples. Nursing bouts are typically brief, lasting only seconds to minutes, as both mother and calf must return to the surface to breathe. Protection from milk dilution is achieved through the forceful ejection system and the seal formed by the calf's mouth. Any water that does enter the mouth is swallowed along with the milk and poses no immediate danger. The high fat content of the milk provides concentrated energy in a small volume, reducing the time required for nursing and minimizing exposure to cold water for the calf. Developmental timeline changes reflect the need for precocial birth. Newborn marine mammals are highly precocial at birth and are already equipped in many ways for an aquatic lifestyle. Harbor seal pups are extremely precocial at birth, already possessing coordinated swimming abilities and the physiological capacity for breath-hold diving. Precocial development of axial locomotor muscle in bottlenose dolphins demonstrates how swimming capabilities are present from birth. The locomotor muscles responsible for powering the tail flukes develop extensively during gestation, allowing newborn calves to swim immediately. Building cetacean locomotor muscles throughout ontogeny supports high-performance swimming into adulthood. The muscles continue developing after birth, but the foundation for swimming is laid during fetal development. Swimming ability from birth is essential for survival. To go for a swim or not has consequences for neonatal aquatic dispersal behavior and growth in gray seal pups. Pups that enter water earlier and swim more extensively show different growth patterns than those remaining primarily on land during early life. Contrasting offspring dependence periods and diving development rates occur even in closely related marine mammal species. Rapid infant development for survival involves multiple coordinated changes. The calf must quickly develop stronger swimming muscles, increase its diving capacity through physiological changes including elevated hemoglobin and myoglobin, develop thicker blubber for thermoregulation, and refine its breathing coordination. These changes occur over weeks to months, representing exceptionally rapid postnatal development compared to terrestrial mammals. Thermoregulation in newborns presents significant challenges. Blubber development in utero versus post-birth affects the newborn's ability to maintain body temperature. The ontogenetic changes in the thermal properties of blubber from fetuses through subadults show that blubber from fetuses through subadults is less conductive than that of adults. The conductivity of blubber from pregnant females is similar to non-adult categories. Blubber from subadults and pregnant females has the highest insulation values, while fetuses and emaciated animals have the lowest. The thermal conductivity of dolphin blubber, which is independent of thickness, remains stable across developmental stages. These results suggest that neonatal blubber is not specialized to provide enhanced insulation, but rather that fetal, neonatal, and juvenile life history stages represent a period of continual blubber growth. Blubber deposition during ontogeny in free-ranging bottlenose dolphins shows continuous accumulation throughout early life. For humans evolving aquatic reproduction, numerous changes would be required. The human birth canal would need to widen substantially to accommodate larger, more developed offspring. Human infants would need to be born more precocially, with swimming muscles already developed and the ability to hold their breath instinctively from birth. The umbilical cord would need to break more easily during delivery. Human milk would need to increase dramatically in fat content while decreasing in water content. Mothers would need to develop forceful milk ejection capabilities. The entire reproductive system would need to accommodate aquatic birth, potentially including tail-first presentation if humans evolved propulsive tails. Chapter 10: Feeding Adaptations and Dietary Shifts. The transition from terrestrial omnivory to aquatic carnivory represents one of the most fundamental dietary shifts in mammalian evolution. This transformation requires not just changes to diet itself, but complete restructuring of the feeding apparatus, digestive system, hunting strategies, and sensory systems used to locate prey. Marine mammals have evolved an extraordinary diversity of feeding strategies, from pursuit predation to filter feeding to bottom foraging, each requiring specialized adaptations. Understanding these changes provides insight into what would be required for humans to successfully exploit marine food resources. Tooth modifications across marine mammal evolution reflect varied dietary strategies. Feeding in marine mammals represents an integration of evolution and functional morphology. The feeding apparatus of marine mammals is driven by developmental changes in the morphology and growth of the skull and teeth. Molecular evolution of tooth-related genes provides insights into dietary adaptations. Mammalian teeth-related genes evolved with different evolutionary histories due to various feeding habits. The evolution of aquatic feeding in seals shows how dental adaptations facilitate underwater prey capture. The development of pierce feeding and loss of oral processing represented major adaptations for underwater feeding in marine mammals. Unlike terrestrial carnivores that capture prey and then chew it extensively, marine mammals typically grasp prey with their teeth and swallow it whole or in large chunks. This strategy avoids the problem of trying to chew underwater, where water would wash food out of the mouth and interfere with mechanical processing. Evolution in carnivorous mammals shows that tooth adaptations evolved in carnivore lineages to cut food into chunks small enough to swallow. The unique pattern of dietary adaptation in the dentition of carnivora demonstrates how carnivorans acquired novel dental variability that benefits their dietary divergence. The carnassial teeth, specialized shearing teeth found in terrestrial carnivores, underwent modifications in marine lineages. Some pinnipeds retained elaborate carnassials for processing prey, while others simplified their dentition to simple pegs suitable only for grasping slippery fish. Carnivore dental adaptations exhibit significant diversity correlating with varied diets across the order. 4 dietary categories are defined based on tooth morphology: hypercarnivores with teeth specialized for meat, mesocarnivores with mixed adaptations, omnivores with more generalized teeth, and hypocarnivores with teeth adapted for non-meat foods. Marine carnivores show varying degrees of specialization depending on their specific prey and feeding strategies. Cetacean dental evolution from terrestrial ancestors followed a distinct trajectory. Pachysteus teeth were intermediate between ancestral and modern states, showing characteristics of both terrestrial carnivores and aquatic hunters. As cetaceans became increasingly aquatic, their teeth simplified into simple conical structures suitable for grasping fish, but not for processing food. The molars lost their complex cusps, becoming single-pointed or even disappearing entirely in some lineages. Baleen evolution in mysticete whales represents one of the most extraordinary feeding innovations in vertebrate history. A bizarre new toothed mysticete from Australia refutes the notions that all stem mysticetes were filter feeders, and that the origins and initial radiation of mysticetes was linked to the evolution of filter feeding. Mysticetes evidently radiated into a variety of disparate forms and feeding ecologies before the evolution of baleen or filter feeding. The phylogenetic context indicates that basal mysticetes were macrophagous predators that did not employ filter feeding or echolocation, and that the evolution of characters associated with bulk filter feeding was. Dynamic filtration in baleen whales involves recent discoveries that have greatly improved our understanding of mysticete oral filtration and have upended the traditional view of baleen filtration as a simple passive process. A second level of complexity arises from differential employment of baleen filters during feeding, which varies widely by taxa. Different mysticete families employ wholly different foraging strategies. Right whales skim continuously through patches of zooplankton, gray whales bottom feed on benthic organisms, and rorquals like blue whales and humpbacks perform lunge feeding on dense prey aggregations. Jaw structure modifications for efficient prey capture occur across marine mammal lineages. The skull reshapes to optimize hydrodynamics while accommodating specialized feeding structures. In cetaceans, the jaws elongate into a beak-like rostrum that reduces drag and allows rapid strikes at prey. In pinnipeds, the jaws remain relatively short but powerful, capable of generating strong bite forces to grasp struggling fish or crush hard-shelled prey. Tongue modifications for underwater feeding include changes to size, shape, and musculature. Baleen whales have enormous tongues that play active roles in filter feeding, helping to force water out through the baleen plates while retaining prey items inside the mouth. In toothed whales, the tongue is less mobile, but still assists in manipulating prey and directing it toward the throat for swallowing. Swallowing mechanisms without aspiration require precise coordination to prevent water from entering the airways while consuming prey underwater. Marine mammals have evolved separated respiratory and digestive pathways that allow them to swallow prey items underwater without any risk of drowning. The larynx forms a tight seal with the nasopharynx, creating a continuous air passage that bypasses the mouth entirely. Digestive system changes accommodate the marine diet and feeding strategies. Stomach compartmentalization in cetaceans creates a complex multi-chambered stomach similar to ruminants, though serving different functions. The first chamber stores recently swallowed prey. The second chamber performs the primary digestion with powerful acid and enzymes, and additional chambers continue the digestive process. This system allows marine mammals to consume large meals rapidly while at prey patches, and then digest the food over extended periods. Intestinal length modifications reflect dietary changes. Carnivorous marine mammals generally have relatively short intestines compared to terrestrial herbivores, as meat protein is more easily digested than plant cellulose. However, intestinal length varies among marine mammals based on their specific diets. Species consuming whole fish, including bones, need longer, more robust intestines than those consuming soft-bodied prey like squid. Enzyme adaptations for marine prey include modifications to digestive enzymes that efficiently process the specific proteins, lipids, and other nutrients found in marine organisms. The high fat content of many marine prey requires elevated lipase production for fat digestion. Salt processing from seawater and marine prey requires specialized mechanisms throughout the digestive tract. Prey detection methods vary dramatically across marine predators. One of the most widespread modes of detection comprises chemical cues, with many predators locating prey items by chemical cues. Within marine environments, prey detectability via visual or chemical cues is affected by environmental conditions such as water clarity and tidal flow. Specialist predators synchronize their foraging activity with cyclic variation in environmental conditions to maximize prey detection. Tracking wakes demonstrates the nocturnal predatory strategy of some aquatic predators. Swimming fish leave wakes containing hydrodynamic and chemical traces that mark their swim paths and could guide predators. Nocturnal European catfish locate piscine prey by accurately tracking its three-dimensional swim path before an attack in the absence of visible light. These results demonstrate that prey wakes remain sufficiently identifiable to guide predators and to extend considerably the area in which prey is detectable. Vision-based hunting remains important for many marine mammals despite the challenges of underwater optics. Clear water environments allow visual detection of prey at substantial distances. Many marine mammals hunt during daylight when visual cues are most reliable. Prey movement attracts visual attention, with predators keying on the flashing silver of schooling fish or the distinctive swimming patterns of different prey species. Echolocation for prey finding provides toothed whales and dolphins with a sophisticated biosonar system. This allows detection of prey in murky water, at night, or at depths where light does not penetrate. Echolocation provides detailed information about prey size, shape, texture, and internal structure. Dolphins can discriminate between different fish species based on their echolocation returns, selecting preferred prey even when multiple species are present. Electroreception in some species, while not widespread among marine mammals, has been documented in certain dolphins. The Guiana dolphin possesses electroreceptive capabilities that allow detection of electric fields generated by living prey. This sense supplements vision and echolocation for prey detection in environments where these other senses are limited. Diving depth capabilities for accessing prey determine the ecological niche available to marine mammals. Shallow divers exploit prey in the photic zone where sunlight penetrates. Deep divers can access prey in the mesopelagic and bathypelagic zones, including deepwater fish, squid, and other organisms unavailable to shallow divers. The deepest diving mammals, including sperm whales and beaked whales, regularly reach depths exceeding 1,000 meters and can dive to over 2,000 meters. Pressure tolerance at feeding depths requires physiological adaptations beyond those needed simply to hold one's breath. The crushing pressure at depth compresses air spaces, increases nitrogen solubility in tissues, and creates mechanical stresses on the body. Marine mammals have evolved flexible rib cages that compress without injury, lung collapse mechanisms that prevent nitrogen absorption, and cellular adaptations that maintain function despite extreme pressure. For humans evolving aquatic feeding adaptations, the dietary shift would likely proceed gradually. Early stages might involve increased consumption of coastal resources, including shellfish, fish, and marine mammals captured in shallow water. This would require relatively modest anatomical changes, perhaps simplified dentition for grasping slippery prey and modifications to digestive enzymes for processing marine proteins and fats. Middle stages would involve pursuit of more mobile prey in deeper water, requiring improved swimming capabilities, enhanced breath-hold endurance, and sensory adaptations for prey detection underwater. Late stages might involve sophisticated specializations such as echolocation for hunting in darkness, extreme diving capabilities for accessing deepwater prey, or potentially even filter-feeding structures for consuming zooplankton, though this would represent a radical departure from typical human omnivory. Chapter 11: Osmoregulation and Kidney Adaptations. Living in a marine environment presents one of the most fundamental physiological challenges for air-breathing mammals, managing water and salt balance in an ocean where the salinity exceeds that of blood and tissues. Terrestrial mammals evolved in environments where fresh water was readily available for drinking, and their kidneys are designed to conserve salts while excreting excess water. Marine mammals face the opposite problem, constantly exposed to water that would draw moisture from their tissues through osmosis, while simultaneously introducing excess salt if consumed. The evolutionary solutions to these challenges involve sophisticated modifications to kidney structure and function, metabolic water production, and behavioral strategies for managing hydration. Osmotic challenges in marine environments stem from the concentration gradient between seawater and body fluids. Seawater contains approximately 1,000 mosmol per liter of dissolved salts, primarily sodium and chloride, while mammalian blood plasma is maintained at approximately 300 mosmol per liter. This threefold difference creates powerful osmotic forces. If a marine mammal's tissues are exposed to seawater, water tends to flow out of the tissues toward the more concentrated seawater, causing dehydration. Conversely, if seawater is consumed, the excess salt must be eliminated without losing precious water in the process. Unlike seabirds and marine reptiles, marine mammals lack specialized glands to excrete salts. Seabirds possess salt glands near their eyes that can excrete highly concentrated brine, and marine iguanas have similar glands in their nasal passages. These specialized structures allow efficient salt elimination with minimal water loss. Marine mammals, however, rely entirely on their kidneys for salt excretion. This places extraordinary demands on the renal system, requiring kidneys capable of producing urine more concentrated than seawater itself. The kidneys of pinnipeds and cetaceans are reniculate in structure, unlike those of most terrestrial mammals except bears. A reniculate kidney consists of multiple discrete lobes called reniculi, each functioning as a miniature kidney with its own cortex and medulla. This architectural difference was once thought to confer greater concentrating ability, but research has shown that reniculation per se does not enhance osmorgulatory capacity. Rather, the key adaptation lies in the thickness and cellular organization of the renal medulla, the inner portion of each reniculus responsible for concentrating urine. Marine mammal kidney specializations include adaptively thickened medullas that provide enhanced ability to concentrate excretory salts in urine. Humans possess lobulated, multipyramidal kidney medullas, a unique attribute among primates. This structural feature is common in aquatic and marine mammals, suggesting it may represent a retained ancestral trait or a convergent adaptation. The significance of this anatomical arrangement is that it increases the length and surface area of the loops of Henle, the tubular structures responsible for establishing the osmotic gradients that concentrate urine. Pinnipeds, cetaceans, manatees, and sea otters can concentrate their urine above the concentration of seawater, but only pinnipeds and otters have been shown to produce urine concentrations of sodium and chloride that are similar to those in seawater. Cetaceans can produce highly concentrated urine in terms of total osmolality, but the sodium and chloride concentrations may not quite reach seawater levels. This suggests that cetacean kidneys concentrate other solutes, particularly urea, to achieve high total osmolality while maintaining sodium and chloride at somewhat lower levels. The glomerular kidney development in saltwater environments shows evolutionary optimization for marine life. The glomerular kidney originally evolved in marine environments as an adaptation for salt excretion and represents a pre-adaptation for freshwater and terrestrial life. However, mammalian kidneys are metabolically inefficient, consuming approximately 80% of available oxygen for sodium reabsorption. This high metabolic cost reflects the fundamental challenge of maintaining precise ion balance while eliminating wastes. Urine concentration capabilities vary among marine mammals based on their ecology and phylogenetic history. Studies assessing the effects of ingested freshwater or seawater in bottlenose dolphins reveal sophisticated osmorgulatory responses. Freshwater ingestion increased urine flow rate while decreasing plasma and urine osmolality, sodium and chloride urine concentrations, and solute excretion rates. This demonstrates that dolphins can efficiently handle freshwater intake by rapidly excreting the excess water while retaining salts. Seawater ingestion resulted in increased urine flow rate, elevated sodium, chloride, and potassium urine concentrations, increased sodium excretion rates, and higher urine osmolality. These changes show that dolphins respond to salt loading by increasing urine production and concentrating salts in the urine. Seawater with protein ingestion was associated with even greater increases in urine flow rate, plasma osmolality, sodium excretion, and urinary solute concentrations. This suggests that protein metabolism produces additional osmotic load that further challenges osmoregulation, and ingestion of protein with seawater appears to further push osmoregulation limits in dolphins. In conclusion, bottlenose dolphins appear to maintain water and plasma solute balance after ingesting fresh water or seawater by altering urine osmolality and solute clearance. This demonstrates remarkable kidney flexibility, capable of responding to diverse hydration challenges. Terrestrial mammals, including humans, generally cannot achieve such dramatic shifts in kidney function. Salt excretion mechanisms in marine mammals operate through enhanced tubular transport systems within the kidney. The loops of Henle and collecting ducts actively transport sodium, chloride, and other ions, creating concentration gradients that draw water from the urine back into the bloodstream. In marine mammals, these transport systems are more powerful than in terrestrial mammals, allowing water reabsorption even when urine already contains high salt concentrations. This countercurrent multiplication system achieves urinary concentrations that would be impossible for human kidneys. Respiratory salt excretion represents a minor pathway for salt elimination in marine mammals. Some salt may be lost through water vapor in exhaled breath, but this route contributes minimally to overall salt balance. The vast majority of excess salt must be eliminated through the kidneys in urine. Drinking seawater and its processing remains controversial in marine mammal physiology. Do whales drink salt water? Indeed, the kidneys of marine mammals are so effective that the latter are capable of excreting a urine that has a higher salt concentration than the water that they drink. However, whether cetaceans routinely drink seawater or obtain most of their water from prey and metabolic production remains debated. Observations suggest that some marine mammals do drink seawater, at least occasionally, and can process it without ill effects thanks to their superior kidney function. Humans cannot drink salt water because their kidneys cannot produce urine more concentrated than seawater. When humans consume salt water, the kidneys must use precious freshwater to dilute the salt for excretion, resulting in net water loss that accelerates dehydration. So ironically, drinking salt water only makes you thirstier. Cetaceans do not have this problem because their kidneys can cycle it more efficiently, producing urine with much higher salt concentration rather than just wasting water to flush it out as fast as possible. Water extraction from prey provides an important water source for marine mammals. Fish and squid contain substantial water within their tissues, typically 60 to 80% water by mass. When marine mammals consume prey, they ingest this water along with proteins, lipids, and other nutrients. The ingested prey water has lower salt concentration than seawater, providing partially desalinated water. However, prey also contains salts dissolved in its tissues, so consuming prey does introduce some salt load that must be managed. Metabolic water production generates additional water through the oxidation of nutrients. The primary source of metabolic water is fat, which generates 1.1 milliliters of water per gram of fat oxidized. Glycogen generates 0.55 milliliters per gram, but glycogen is hydrated and releases an additional 2 to 3 grams of water upon breakdown, making it a more significant source of water despite lower energy density. Protein catabolism produces some metabolic water, though muscle tissue yields water primarily from its 70% water content rather than from metabolic oxidation. Because fat stores can be massive in marine mammals, whereas glycogen stores are limited, these animals generate most of their metabolic water from fat rather than glycogen. Blubber serves not just as insulation and energy storage, but also as a water reservoir that can be tapped during periods when prey is scarce or when fasting during migration or reproduction. For desert rodents that also rely heavily on metabolic water, the fat is often stored in the tail to minimize heat gain. Marine mammals distribute fat across the body as blubber, serving thermal as well as metabolic needs. However, the problem with generating metabolic water from fat is that there is a relatively higher requirement for oxygen, which necessitates increased respiration. Because the lungs humidify expelled air, there is minimal net water gained under normal conditions where evaporative water loss is high. However, in settings where evaporative water loss is minimized, the generation of metabolic water can be substantial. For marine mammals breathing through blowholes with minimal exposed respiratory surface, evaporative losses are reduced compared to terrestrial mammals, making metabolic water production more efficient. Vertebrates have expanded their habitats during evolution, which accompanies diversified routes for water acquisition. Water is acquired by oral intake and subsequent absorption by the intestine in terrestrial and marine animals, which are subjected to constant dehydration. Experiments using eels suggest that water production by lipid metabolism is an additional route for water acquisition when they encounter seawater. It seems that metabolic water production contributes to counteract dehydration before mechanisms for water regulation are reversed from excretion in freshwater to acquisition in seawater. Water conservation strategies employed by marine mammals include behavioral and physiological mechanisms. Reduced urination frequency compared to terrestrial mammals means that marine mammals produce concentrated urine less often, conserving water between urination events. Highly concentrated urine production allows maximum salt excretion with minimum water loss, achieving ratios impossible for terrestrial mammals. The thick medulla and elongated loops of Henle create the osmotic gradients necessary for this extreme concentration. Blood iron regulation must maintain precise sodium, chloride, potassium, calcium, magnesium, and other iron concentrations despite varying salt intake and water availability. Sodium and chloride balance is particularly critical given their abundance in seawater and their importance for nerve and muscle function. Potassium management requires careful regulation as both deficiency and excess can be life-threatening. Calcium and magnesium handling involves coordination between kidneys, bones, and intestines to maintain appropriate levels for cellular function. Hormonal control of water balance involves multiple endocrine systems. Antidiuretic hormone, also called vasopressin, is modified in marine mammals to enhance water retention when needed. This hormone increases water reabsorption in the kidney collecting ducts, concentrating urine. Aldosterone function in marine mammals regulates sodium retention and potassium excretion, fine-tuning electrolyte balance. Atrial natriuretic peptide roles include promoting sodium excretion when salt loads are excessive, counterbalancing the sodium-retaining effects of aldosterone. For humans evolving toward aquatic life, osmorgulatory adaptations would develop gradually over hundreds of thousands to millions of years. Early changes might include thickening of the renal medulla and elongation of the loops of Henle, modestly improving urine concentrating ability. These relatively simple anatomical changes could appear within tens of thousands of generations. The existing lobulated kidney structure in humans might represent a pre-adaptation that could be elaborated, providing a starting point for further modification. Enhanced hormonal sensitivity and modified receptor densities would improve the kidney's ability to respond to varying hydration states. More dramatic changes, including the ability to produce urine significantly more concentrated than seawater and the metabolic reorganization to efficiently generate water from fat oxidation while minimizing respiratory water loss, would require much longer evolutionary periods. Complete osmorgulatory independence from fresh water, allowing permanent residence in marine environments without ever needing to drink fresh water, represents the end point of this evolutionary trajectory and might require millions of years to achieve fully. Chapter 12: Neural and Cognitive Changes. The brain orchestrates all complex behaviors, processes sensory information, generates consciousness, and enables learning, memory, and social interactions. For aquatic mammals, the cognitive demands of marine life have driven remarkable brain evolution. Three-dimensional navigation through featureless ocean, echolocation for hunting and orientation, sophisticated social structures, extended parental care, and cultural transmission of knowledge all require substantial neural processing power. Understanding how cetacean brains evolved and function provides insight into the neural changes that would accompany any human transition to aquatic life. Brain size and structure in marine mammals show extraordinary diversity and sophistication. The encephalization quotient, defined as the ratio between actual brain mass and predicted brain mass for an animal of a given size, provides a measure of relative brain size across species. The average encephalization quotient is 1.0, while the encephalization quotient value for humans is 7.0. This means that human brains are 7 times the size that would be expected for a species of the same average body weight. Humans have the highest encephalization quotient at 7.4, but bottlenose dolphins have encephalization quotients of 5.3. This places dolphins second only to humans in relative brain size among all mammals. Dolphin and whale intelligence demonstrates remarkable cognitive abilities. Laboratory studies of bottlenose dolphins have documented various dimensions of their intellectual abilities, including understanding of symbolic representations of things and events, declarative knowledge, understanding of how things work or how to manipulate them, procedural knowledge, understanding of the activities, identities, and behaviors of others, social knowledge, and understanding of one's own image, behavior, and body parts, self-knowledge. All these capabilities rest on a strong foundation of memory, with investigations demonstrating that bottlenose dolphin auditory, visual, and spatial memory are accurate and robust. The cetacean neocortex surpasses in gyrification all other mammals, including humans. Gyrification refers to the folding of the brain surface, with more folds creating greater surface area within a given skull volume. The extensive folding of cetacean brains indicates massive expansion of cortical tissue. The cetacean neocortex comprises limbic, paralimbic, and supralimbic regions, showing regional specialization similar to other mammals but organized along different architectural principles. Cetacean brain structure complexity reveals unique organizational features distinct from terrestrial mammals, whereas subcortical neuroanatomy is shared across mammals with variations mainly in the proportions of various structures. The massive expansion of the cetacean cortex occurred along a very different organizational route than that of other mammal brains, including humans. The cetacean neocortex was once viewed as relatively homogeneous in cellular architecture, regionally unspecialized, and lacking organizational complexity. However, recent research has completely overturned this outdated view, revealing sophisticated cortical organization with distinct functional areas. Recent imaging studies show that it is precisely by a greater proportion of white matter that humans can be distinguished from apes and monkeys. White matter consists of myelinated axons connecting different brain regions, enabling rapid long-distance communication within the brain. Growing evidence demonstrates that astrocytes, supporting cells in the brain, contribute to the modulation and coordination of neural activity. Therefore, despite earlier arguments, a high glial cell-to-neuron ratio is consistent with the increased needs of complex brains for rapid communication and synaptic efficiency. Cetaceans and primates show convergence in intelligence and self-awareness despite their brains being organized along different lines. Both groups exhibit self-recognition in mirrors, complex social behaviors, cultural transmission of knowledge, and sophisticated problem-solving abilities. This cognitive convergence suggests that intelligence can evolve through multiple neural architectures, each arriving at similar functional outcomes through different structural organizations. DHA and brain evolution connections provide crucial context for understanding how diet influences neural development. Docosahexaenoic acid is the most abundant omega-3 fatty acid in the human brain. An important turning point in human evolution was the discovery of high-quality, easily digested nutrients from coastal seafood and inland freshwater sources. Compelling evidence suggests that the discovery and subsequent multi-generational exploitation of seafood coincides with the rapid expansion of the cerebral cortex that is unique to modern humans. The brains of 42 modern mammalian species were studied and found to be similar in brain chemistry, particularly in the predominant use of DHA in the membrane-rich neural tissues at synapses and in the retina. This universality of DHA in neural tissue across mammals suggests its fundamental importance for brain function. A marine food diet high in critical brain-building nutrients, especially DHA, phospholipids, and vitamin B12, is the best explanation for cerebral expansion in human evolution. Docosahexaenoic acid, the omega-3 fatty acid found in large amounts in seafood, boosts brain growth in mammals. The aquatic diet hypothesis for brain evolution proposes that marine food chains were necessary to provide sufficient DHA for human brain expansion. Whether access to aquatic foods, particularly those rich in docosahexaenoic acid and other long-chain polyunsaturated fatty acids, was necessary for the evolution of the large human brain, remains actively debated. Some researchers argue that marine food chain access was essential, while others contend that terrestrial food sources could provide adequate nutrition. Recent studies found that African lake fish could supply enough DHA to potentially kick off human brain evolution, but not enough for the rapid growth seen in later stages. DHA accretion rates in fetal development show that 14.6 milligrams per week are required before birth compared to 5.5 milligrams per week after birth. This demonstrates the critical importance of maternal DHA intake during pregnancy for fetal brain development. Seafood boosting brain growth in mammals extends beyond humans, with dolphins showing remarkably large brains relative to body size. Cognitive demands of aquatic life drive neural adaptations. Three-dimensional spatial navigation requires fundamentally different cognitive processing than two-dimensional terrestrial navigation. Fish have a higher-order representation of space that assembles incoming sensory information into a neural unit that can be used to determine position and heading in 3 dimensions. This representation appears to be encoded isotropically, as would be expected for animals that move freely through volumes. The neural basis of spatial representation involves interconnected brain structures that encode both distance and direction. The representation of 3-dimensional space in fish reveals that spatial cognition is not limited to mammals, but represents a deep-seated vertebrate trait. Fish were able to extract the horizontal component from a learned 3-dimensional path, revealing that they had learned and remembered spatial information from both dimensions. Similar capabilities would be required for humans evolving toward fully aquatic life, necessitating neural modifications to process and remember 3-dimensional spatial information. Echolocation neural processing requires dedicated brain regions for analyzing acoustic returns. Changes in cranial architecture and ear structure to support echolocation appeared relatively early in cetacean evolution. The neural circuitry for echolocation must extract information about object distance, size, shape, texture, and movement from temporal and spectral patterns in returning echoes. This demands sophisticated signal processing capabilities and likely involves expansion of auditory cortex regions specialized for analyzing complex sounds. Social complexity in marine mammal groups drives cognitive evolution. Understanding the activities, identities, and behaviors of others, termed social knowledge, requires substantial neural processing. Dolphins must recognize individual pod members, remember past interactions, predict others' behaviors, and coordinate group activities, including cooperative hunting. These social cognitive demands likely contributed to the expansion of cetacean brains. Communication system evolution includes sophisticated acoustic signaling. Cetaceans are the next frontier for vocal rhythm research, with evidence showing that rhythm, particularly isochronous rhythm, when sounds are equally spaced in time, is prevalent in cetacean vocalizations. Quantitative evidence of rhythm has been found in 34 vocalizations from 23 cetacean subspecies. This likely underestimates the actual diversity, given that few studies have explicitly quantified rhythm in cetacean vocalizations. Vocal communication is the main means of communication for cetaceans. Acoustic communication is most important for cetaceans, as sounds, out of all signal forms, propagate best in the water environment. Just like the species themselves, their repertoires are diverse, consisting of tonal, pulsed, and combined calls, with frequency and amplitude modulation. Long-distance communication in ocean water allows whales to potentially communicate across hundreds of kilometers using low-frequency vocalizations that travel efficiently through water. Neural adaptations for breath-hold diving include modifications protecting the brain from hypoxia. Hypoxia tolerance at cellular level involves enhanced ability of neurons to maintain function despite reduced oxygen availability. Neuroprotection mechanisms include elevated brain globins, neuroglobin, and cytoglobin that store and transport oxygen within neural tissue. Cerebrovascular adaptations in free-divers include modifications to blood vessels supplying the brain that maintain blood flow and oxygen delivery during dives. Brain oxygen prioritization through selective vasoconstriction ensures that even during extreme dives, cerebral blood flow is maintained. Sleep pattern modifications in cetaceans include unihemispheric slow-wave sleep, one of the most remarkable neural adaptations. Cetaceans, or whales and dolphins, have a pretty unique way of getting some shut-eye by resting only one half of their brain at a time. While one hemisphere of the brain rests, the other remains alert. The hemispheres are alternated between sleep and consciousness a number of times in each resting period, which can last anywhere between a few minutes and a few hours. Dolphins sleep with one eye open and with half their brain wide awake. The unihemispheric slow-wave sleep, the ability to sleep during swimming with one open eye, and the absence of paradoxical sleep in its form observed in all terrestrial mammals are unique features of sleep in cetaceans. It was suggested that the main adaptive functions of unihemispheric sleep consist in combination of sleep and movement, as well as of sleep and sensory control of the environment state. Continuous breathing awareness necessitates some level of consciousness maintained at all times, as cetaceans must consciously surface to breathe. Unlike terrestrial mammals, where breathing is largely automatic even during sleep, cetaceans cannot afford complete unconsciousness. Unihemispheric sleep solves this problem by keeping one hemisphere awake to monitor breathing needs and surface position while allowing the other hemisphere to rest. For humans evolving toward aquatic life, neural and cognitive changes would unfold over extended evolutionary periods. Enhanced three-dimensional spatial processing would develop relatively early, within hundreds of thousands of years, as navigation in marine environments immediately benefits from improved volumetric spatial cognition. If echolocation evolved, which would require millions of years, substantial expansion of auditory processing regions would be necessary, along with development of entirely new neural circuits for echo analysis. Unihemispheric sleep might emerge over hundreds of thousands of years as the need for conscious breathing control became increasingly important during extended aquatic periods. Chapter 13. Timeline of Transformation Generation by Generation. Understanding the realistic timescale for human aquatic evolution requires examining both the fossil evidence from other mammalian transitions and the fundamental mechanisms of evolutionary change. Evolution operates through natural selection, acting on heritable variation across generations, and the rate at which populations adapt depends on multiple factors, including selection pressure intensity, genetic variation availability, population size, and the complexity of the traits being modified. By combining insights from whale evolution, recent human evolutionary changes, and population genetics theory, we can construct a realistic timeline for how humans might gradually transform from terrestrial to fully aquatic mammals. The cetacean evolutionary timescale provides our primary benchmark for understanding how long terrestrial to aquatic transitions require. Whales evolved from their oldest terrestrial ancestor to their most recent aquatic ancestor over the course of about 14 million years. The first whales appeared 50 million years ago, well after the extinction of the dinosaurs, but well before the appearance of the first humans. Their ancestor is most likely an ancient artiodactyl, a four-legged, even-toed hoofed land mammal adapted for running. Whale evolution represents a rapid transition from land to sea in geological terms. The evolutionary sequence from Pachycetus to modern cetaceans documents this transition. Pachycetus lived approximately 50 million years ago. This wolf-sized terrestrial carnivore possessed characteristics identifying it as an early whale ancestor despite its fully terrestrial lifestyle. The fossil of Pachycetus was found in Pakistan in 1978, revealing a creature that spent most of its time on land but likely hunted along coastlines and in shallow waters. Ambulocetus emerged approximately 49 to 47 million years ago. The Caldana Formation is constrained to sometime during the Lutetian stage of the early Eocene, and the remains may date to 48 to 47 million years ago. This evolved version of Pachycetus improved greatly. Ambulocetus was about 10 feet long and weighed 500 pounds. Its snout was narrower, it had webbed feet, and a longer tail for swimming. Its diet consisted of fish and crustaceans. It was still a freshwater whale. Ambulocetus was the walking and swimming whale, and its marine ancestor still lived primarily on land. This represents approximately 1 to 3 million years of evolutionary change from Pachycetus. Rhodhostis appeared approximately 46 million years ago. In 1994, fossil hunters found a 46 million-year-old fossil skeleton on the shores of Central Asia. This evolved version of Ambulocetus had a shorter head, longer legs with flippers, and a wider tail for better swimming. Its diet consisted of fish and squids. Rhodhostis spent most of its time in the water rather than on land, although it was still capable of walking on land. As the sea became shallower and much more fish were in the sea, Rhodhostis started to hunt in salt water. This represents approximately 2 to 3 million years from Ambulocetus. Maiacetus lived approximately 47 million years ago, contemporary with Ambulocetus, but showing different adaptations. This species was amphibious with teeth like toothed whales. Dorudon existed 34 to 40 million years ago. This species showed a blowhole, flippers, reduced hind limbs, flukes, and was fully aquatic. Lanostus lived 33.9 to 38 million years ago. This primitive baleen whale was fully aquatic with an immobile elbow and flukes. These dates reveal that the transition from terrestrial Pachycetus to fully aquatic forms like Dorudon took approximately 10 to 16 million years. The early stages, moving from fully terrestrial to semi-aquatic amphibious forms, occurred within the first 5 million years. The later stages, achieving fully aquatic specialization with complete loss of terrestrial capabilities, required an additional 5 to 10 million years. Human generation time provides the conversion factor from years to generations. Human generation times across the past 250,000 years show an average of 26.9 years. Analyses of whole genome data reveal an average generation time of 26.9 years across the past 250,000 years, with fathers consistently older at 30.7 years than mothers at 23.2 years. For simplicity, we can use approximately 27 years per generation, or roughly 37 generations per 1,000 years. Calculating required generations for various adaptations allows us to project realistic timescales for human aquatic evolution. If the full terrestrial to aquatic transition requires 10 million years at minimum, this represents approximately 370,000 generations. If we compress this timeline, assuming stronger selection pressures or more available genetic variation than cetaceans experienced, we might achieve the transition in 5 million years, representing approximately 185,000 generations. These numbers are staggering but necessary for truly comprehensive evolutionary transformation. Selection pressure intensity needed determines how rapidly evolution can proceed. Rapid evolutionary adaptation in response to selection can occur under certain circumstances. The mechanisms facilitating rapid adaptation range from strong positive directional selection leading to large shifts in the allele frequencies at a few loci, called selective sweeps, to polygenic selection causing small changes in allele frequencies at many loci. Evolutionary adaptation after sudden environmental changes can occur very rapidly when selection is intense. Limits on the evolutionary rates of biological traits exist based on fundamental physical and biological constraints. A geometric speed limit for acceleration by natural selection in evolutionary dynamics restricts how quickly populations can adapt. This speed limit derives from information theoretic principles and places fundamental bounds on evolutionary rates regardless of the specific biological system. Remarkably, the selection bound can be much tighter if the contribution of selection is more dominant than that of mutation. Natural versus artificial selection scenarios differ dramatically in selection intensity. Natural selection operates through differential survival and reproduction, with selection coefficients typically ranging from 0.01 to 0.1, meaning 1 to 10% differences in fitness. Artificial selection imposed by human breeding programs can achieve much stronger selection, with coefficients approaching 1.0, meaning complete selection in a single generation. For human aquatic evolution, we assume natural selection operating in the wild without artificial intervention. Environmental pressures driving adaptation would need to be substantial and sustained over many generations. Climate change creating coastal dependence represents one plausible scenario. Rising sea levels are causing unprecedented changes in coastal habitats, with impacts on marine ecosystems worldwide. Rising sea levels are forcing marine species to adapt at rates faster than ever previously recorded. This rapid change affects everything from small invertebrates to large marine mammals, disrupting feeding patterns, breeding grounds, and migration routes established over millennia. Sea level rise driven by thermal expansion and melting ice threatens coastal populations. The oceans take up more than 90% of the excess heat trapped by greenhouse gases. As ocean temperatures rise, this absorbed heat leads to the expansion of water, contributing significantly to sea level rise. Melting of glaciers and polar ice sheets releases fresh water into the sea. If sea levels rose dramatically over thousands of years, coastal human populations might be forced increasingly into aquatic lifestyles, creating strong selection for aquatic adaptations. Food scarcity on land driving marine foraging represents another potential selective pressure. If terrestrial food resources declined while marine resources remained abundant, populations increasingly dependent on fishing, diving, and marine mammal hunting would experience selection favoring traits that improve aquatic foraging efficiency. Each generation, individuals with slightly better swimming ability, longer breath holds, improved underwater vision, or other aquatic advantages would tend to have more offspring, gradually shifting population trait distributions. The first 100,000 years, representing approximately 4,000 generations, would see relatively modest changes building on existing human capabilities. Enhanced diving reflex intensity would increase, with heart rate dropping more rapidly and dramatically upon face immersion. Individuals with stronger diving reflexes would sustain longer breath holds and dive deeper for food, providing fitness advantages. Increased lung capacity would develop through genetic changes affecting chest size, rib number, and diaphragm strength. Individuals born with slightly larger lungs could hold more oxygen, stay submerged longer, and access deeper prey. Improved breath-holding time would increase from the current untrained average of 30 to 90 seconds towards several minutes, even without training. Genetic variants affecting carbon dioxide tolerance, oxygen storage, and metabolic rate during breath holds would be strongly favored. The Bajau people demonstrate that measurable genetic adaptations for diving can evolve within perhaps thousands of years. Their 50% larger spleens evolved through natural selection, acting on populations heavily dependent on breath-hold diving for subsistence. Subcutaneous fat increase would begin during this period, with average fat layer thickness increasing from current levels of several millimeters toward 1 to 2 centimeters. This modest increase would provide improved insulation in water and energy storage for extended swimming. Reduced body hair would accelerate, building on the existing human tendency towards sparse body hair. Genetic variants that further reduced hair density and diameter would be favored, as smoother skin reduces drag during swimming. Webbing between fingers and toes would likely appear through mutations affecting developmental pathways. Syndactyly mutations that are currently deleterious in terrestrial humans would become advantageous in aquatic environments. Even partial webbing significantly improves swimming efficiency, providing immediate fitness benefits. Swimming efficiency improvements would accumulate through multiple mechanisms, including better body positioning, more effective stroke patterns, and reduced drag from streamlining. The period from 100,000 to 500,000 years, representing generations 4,000 through 20,000, would show more substantial morphological changes. Significant body hair loss would leave humans nearly hairless across most body surfaces, retaining hair only on the head and perhaps genital regions. Thick blubber layer development would increase subcutaneous fat to 5 to 10 centimeters in thickness, providing substantial insulation and energy storage. The continuous fat layer would begin developing the reinforcing collagen networks seen in pinniped blubber. Streamlined body shape would emerge through selection for reduced drag. The body would become more fusiform, with reduced surface irregularities and protrusions. Shoulders would broaden relative to hips, facilitating more powerful arm strokes. Skin waterproofing would improve through changes to epidermal structure and lipid composition. Enhanced underwater vision would develop through lens modifications, making it more spherical and powerful. The cornea might flatten slightly, and protective structures like enhanced tear production or nascent nictitating membranes might appear. Enhanced diving capabilities would extend breath holds to 5 to 10 minutes for average individuals through accumulated changes to blood oxygen capacity, myoglobin concentrations, and metabolic regulation. Diving to depths of 50 to 100 meters would become routine as pressure tolerance improved and the diving reflex strengthened. Kidney modifications would begin, with medullary thickness increasing and urine concentrating ability improving modestly. From 500,000 to 2 million years, representing generations 20,000 through 80,000, dramatic skeletal and physiological restructuring would occur. Nostril migration would begin, with the external nares moving gradually dorsally and posteriorly on the face and skull. This would require coordinated changes to skull bones, nasal passages, and surrounding soft tissues. Limb transformation to flippers would progress, with fingers lengthening, bones flattening, and arm flexibility reducing at elbow and wrist joints. The hands would become more paddle-like, functioning as integrated swimming appendages rather than manipulative tools. Tail structure development would initiate through elongation of the coccyx and development of new vertebrae. Muscular attachments would strengthen along the lower spine and developing tail. The beginnings of lateral tail flukes might appear as connective tissue expansions from the caudal vertebrae. Major skeletal restructuring would reshape the entire postcranial skeleton for aquatic locomotion. The spine would straighten and lengthen, vertebrae would become more uniform, and the ribcage would expand. Complete hair loss would leave the skin entirely bare, except perhaps the scalp. Advanced echolocation precursors might develop, including modifications to produce rapid clicks and nascent fatty tissue accumulations in the forehead region. Reproductive adaptations for water birth would begin, including modifications to the birth canal, infant development toward more precocial states, and changes to maternal lactation supporting aquatic nursing. From 2 to 5 million years, representing generations 80,000 through 200,000, the transformation would accelerate toward full aquatic specialization. Blowhole formation would complete the nostril migration, positioning the respiratory opening on top of the head. This would require wholesale reorganization of skull architecture and soft tissue pathways. Fully functional flippers would replace the arms, with all joints except the shoulder becoming immobile. The flippers would be flattened, streamlined, and reinforced for efficient propulsion and steering. Powerful tail flukes would develop as the primary propulsive organ. The tail would extend through dozens of additional vertebrae fused into rigid sections. Horizontal flukes several feet across would project laterally, powered by massive muscles along the spine and tail. Streamlined skull would complete the hydrodynamic optimization of the head, with the face projecting smoothly and all irregularities minimized. Deep diving adaptations would enable routine dives to 300 to 500 meters and extended dives to 1,000 meters or deeper. Complex underwater communication would develop through sophisticated vocalizations, potentially including proto-language capabilities mediated by acoustic signals. Beyond 5 million years, the population would achieve complete aquatic specialization, functionally indistinguishable from other marine mammals. Potential loss of terrestrial mobility would occur if hind limbs atrophied or disappeared entirely. Complete physiological commitment to ocean would eliminate the ability to survive on land for extended periods. The respiratory system would require conscious control of breathing at all times, making sleep on land impossible without drowning risk. Genetic mechanisms of change underlying this transformation would operate through multiple pathways. Point mutations and adaptation would provide the raw material for evolutionary change. The mutation rate in human evolution and demographic inference reveals that the germline mutation rate in humans is approximately 0.5 times 10 to the -8th mutations per base pair per generation. This translates to approximately 60 new mutations per individual per generation across the 3 billion base pair human genome. Gene regulation modifications often contribute more to adaptive evolution than changes to protein coding sequences themselves. Many adaptations arise through altered expression timing, expression levels, or tissue-specific expression of existing genes rather than creating entirely new genes. Regulatory evolution can produce rapid adaptive changes because a single regulatory mutation can affect multiple downstream genes simultaneously. Gene loss importance in evolution appears counterintuitive, but proves crucial for aquatic adaptation. Genes lost during the transition from land to water in cetaceans include those regulating hair development, sweat glands, and olfactory receptors. Evolution through gene deletion represents an important mechanism that removes functionalities no longer needed in the new environment, potentially reducing metabolic. Selective sweeps would occur when beneficial mutations arise and rapidly increase in frequency. A selective sweep is the process through which a new beneficial mutation that increases its frequency and becomes fixed in the population. Hard selective sweeps occur when a single new mutation sweeps to fixation. Soft selective sweeps involve multiple independent beneficial mutations at the same locus or standing genetic variation that already existed in the population. For complex aquatic adaptations, soft sweeps from standing variation might be more common than hard sweeps from new mutations. Chapter 14: Social and Behavioral Evolution. Beyond anatomical and physiological changes, the transition to aquatic life would fundamentally reshape human social structures, behaviors, and cultural practices. Marine mammals exhibit remarkably complex social organizations that differ substantially from terrestrial mammals, reflecting the demands and opportunities of oceanic environments. Understanding cetacean and pinniped social systems provides insight into how human social behavior might evolve during aquatic adaptation. Communication, cooperation, parental care, mating strategies, migration patterns, and cultural transmission would all undergo substantial modifications shaped by the marine environment. Social structure changes for aquatic life begin with the basic grouping patterns of individuals. Pod formation offers multiple benefits that would strongly favor group living in aquatic humans. The fundamental unit of cetacean society is the pod, a relatively stable group of related and unrelated individuals that travel, forage, and socialize together. Pod sizes vary enormously across species, from small family groups of 5 to 10 individuals in some dolphins to massive aggregations of hundreds or thousands in some whale species. Protection from marine predators represents a primary advantage of group living. Large sharks, killer whales, and potentially other predators would pose threats to solitary aquatic humans. Group vigilance allows earlier predator detection, with many eyes watching for danger. Coordinated defense through group mobbing or counterattacks makes predators more likely to abandon attacks. Confusion effects from groups make it difficult for predators to single out individual targets. These anti-predator benefits would strongly select for sociality in aquatic humans. Cooperative hunting strategies enable groups to capture prey more efficiently than solitary individuals. Cetaceans demonstrate numerous cooperative hunting techniques, including encirclement, where individuals surround schools of fish and take turns feeding while others maintain the encirclement. Herding prey towards surfaces or coastlines traps prey against barriers, making capture easier. Bubble net feeding in humpback whales creates curtains of bubbles that concentrate prey. Coordinated deep diving in sperm whales may allow groups to exploit prey resources more thoroughly than individuals could alone. Social learning importance increases dramatically in aquatic environments where trial-and-error learning can be costly or fatal. Young individuals must learn to hunt effectively, navigate three-dimensional spaces, locate productive foraging areas, avoid predators, and coordinate with group members. Observing and imitating successful adults provides a safer and faster learning pathway than individual trial and error. Teaching offspring to hunt involves active instruction documented in several cetacean species. Mothers demonstrate hunting techniques to calves, adjusting their behavior to facilitate calf learning. Cultural transmission of techniques allows knowledge to accumulate across generations and spread through populations. Different cetacean populations show distinct hunting strategies, vocal dialects, and social behaviors that are culturally transmitted rather than genetically determined. For example, different killer whale populations specialize on different prey types, fish versus marine mammals, and these specializations are culturally learned and maintained across generations. Aquatic humans would likely develop diverse cultural traditions related to foraging strategies, tool use, and social organization. Dialect development in communication reflects cultural evolution of vocal repertoires. Cetacean populations develop regionally distinct acoustic patterns analogous to human languages and dialects. These vocal traditions are learned rather than innate and can change over time as innovations spread through populations. Aquatic humans would likely maintain complex vocal communication, transitioning from aerial vocalizations to underwater acoustic signals. The development of distinct regional dialects would reflect both cultural drift and adaptive specialization to local environments. Mating system modifications would adapt to the challenges and opportunities of aquatic reproduction. Aquatic courtship behaviors would replace terrestrial courtship, incorporating underwater displays, acoustic signals, and synchronized swimming. Males might compete for females through elaborate vocal performances, physical displays of size and strength, or demonstrations of hunting prowess. Female choice would select for male traits indicating genetic quality, provisioning ability, or social status. Underwater mating mechanics require specific positioning and coordination. Cetaceans typically mate belly to belly, often while swimming, requiring precise control and cooperation. For aquatic humans, similar adaptations would be necessary to accomplish reproduction underwater. Alternatively, mating might occur at the surface or in shallow water, particularly during early stages of aquatic adaptation before fully underwater mating capabilities evolved. Seasonal breeding patterns might develop in response to environmental cycles, affecting food availability or suitable birthing conditions. Many marine mammals show seasonal reproduction, with mating occurring at specific times of year and births concentrated during periods when conditions favor calf survival. For aquatic humans, similar patterns might emerge, with conception occurring when food is abundant to support pregnancy and lactation, and births timed to coincide with warm water temperatures or prey availability. Migration development would become central to aquatic human ecology as populations tracked seasonal resources across vast ocean distances. Following food resources drives migrations in most marine mammals. Baleen whales migrate between polar feeding grounds where productivity is high during summer and tropical breeding grounds where warm waters favor calf survival during winter. Toothed whales and dolphins show more complex movement patterns, but many species undertake seasonal migrations following prey distributions. Breeding ground establishment would create traditional locations where aquatic humans gather for mating and giving birth. These areas would offer specific advantages, including shallow warm water for calves, protection from predators, or abundant food resources. Cultural transmission would maintain knowledge of breeding ground locations across generations, with experienced individuals guiding others to traditional sites. Long-distance navigation across featureless ocean requires sophisticated cognitive mapping and orientation mechanisms. Marine mammals navigate using multiple cues, including sun position, star patterns, Earth's magnetic field, landmarks such as seamounts and coastlines, and acoustic features of the seafloor. Aquatic humans would develop similar capabilities through genetic evolution of sensory systems and cultural transmission of navigational knowledge. Magnetic sense development might emerge if genetic variation affecting magnetoception appeared and was favored by selection. Play behavior in marine mammals serves multiple functions in social and cognitive development. Play among juveniles develops swimming skills, hunting techniques, and social bonds. Common play behaviors include chasing, wrestling, tossing objects, and riding waves. Adult play, also frequent in cetaceans, maintains social relationships, and provides cognitive stimulation. Aquatic humans would likely maintain or even expand play behaviors, given our species' existing strong tendency toward play throughout life. Surfing and wave riding, observed in many cetacean species, combines play with efficient travel. Dolphins frequently ride bow waves of boats and pressure waves from large whales, surfing without expending energy. Wave riding for play and transport would appeal to aquatic humans, combining pleasure with practical benefits. Aquatic humans might also surf breaking waves near coastlines, much like terrestrial humans do recreationally today, but as an integrated part of aquatic lifestyle. Tool use possibilities exist for aquatic humans given our species' extraordinary reliance on technology. Marine mammal tool use examples include dolphins using marine sponges held in their mouths to probe seafloor substrates for hidden prey, a behavior culturally transmitted in specific populations. Sea otters use rocks to crack open shells while floating on their backs, showing considerable dexterity despite flippers. Aquatic humans might develop or maintain tool use for food processing, shelter construction, or hunting enhancement. Cognitive requirements for tool use include understanding cause and effect relationships, planning future actions, and social learning to transmit techniques. Aquatic humans would likely retain high cognitive abilities supporting tool manufacture and use, though modifications to hand structure into flippers would constrain manipulation capabilities. Tools might be simplified compared to terrestrial human technology, but could still include objects for prying open shells, cutting tough prey, or defense against predators. Territorial behavior in water manifests differently than terrestrial territoriality. Some cetaceans defend specific areas as feeding or breeding territories, but boundaries are less fixed than terrestrial territories. Defending hunting grounds might occur if particularly productive areas are localized and defendable. Coral reefs, kelp forests, or coastal upwelling zones could become territorial focal points. Hierarchical social structures would determine access to prime foraging areas, with dominant individuals or coalitions controlling best locations. Aggressive displays underwater would replace terrestrial aggression with aquatic-appropriate behaviors. These might include charging at opponents while vocalizing loudly, jaw-clapping or teeth-baring, tail slaps creating pressure waves, or physical ramming. Most conflicts would likely resolve through displays rather than actual combat, as injuries underwater could prove fatal through drowning, predation, or infection. Interspecies interactions would shape aquatic human ecology profoundly. Competition with existing marine mammals for prey resources would create interspecific tension. Overlapping dietary preferences with dolphins, seals, or small whales could lead to competition. Predator-prey relationships might position aquatic humans as prey for large sharks or killer whales, at least during early evolutionary stages when humans remained relatively small and vulnerable. Conversely, aquatic humans might hunt smaller marine mammals for food, reversing the traditional prey relationship. Symbiotic relationships with fish might develop through mutualistic interactions. Some marine mammals have been observed in apparent cooperative relationships with fish, though these are typically not true symbioses. Aquatic humans might develop clearer mutualistic bonds, perhaps using fish as indicators of prey presence or allowing certain fish species to clean parasites from their skin. Sleep and rest patterns would undergo fundamental modifications, as discussed in the neural evolution section. Sleeping while maintaining buoyancy requires either conscious muscle control to stay at the surface or physiological buoyancy control through air in lungs or fat distribution. Unihemispheric sleep development would allow one brain hemisphere to rest, while the other maintains breathing awareness, orientation, and environmental monitoring. Rest areas would include sheltered coastal locations, floating at the surface in calm water, or potentially drifting with currents while maintaining light sleep. Group dynamics would influence all aspects of aquatic human society. Leadership in pods would emerge through multiple pathways, including physical dominance, reproductive success, knowledge of resources, or social manipulation. Matriarchal structures, common in some cetaceans, might develop if females acquire critical ecological knowledge through long reproductive careers. Alternatively, male-dominated hierarchies might persist if male size and strength advantages remain important for resource competition and predator defense. Altruistic behaviors, including caregiving for injured or sick members, are well documented in cetaceans. Epimelectic behavior, where individuals support injured pod members at the surface to breathe, occurs in multiple species. Similar behaviors would likely persist or strengthen in aquatic humans given our existing strong tendencies toward compassion and caregiving. Helping injured individuals would be critical for survival, as an injured swimmer unable to surface independently would quickly drown without assistance. Allomaternal care, where individuals other than the mother care for offspring, is common in cetaceans and would likely characterize aquatic human societies. Experienced females might assist new mothers with birth and calf care, increasing survival rates. These auntie behaviors would provide learning opportunities for nulliparous females while supporting reproductive success of related individuals. Chapter 15: Ecological Impact and Niche Competition. The emergence of an aquatic human species would not occur in ecological isolation. Every environment contains finite resources that support limited populations, and the introduction of a new apex predator would fundamentally alter marine ecosystems. Understanding the ecological consequences of human aquatic evolution requires examining carrying capacity limits, competitive interactions with existing marine mammals, impacts on prey species, habitat requirements, disease dynamics, and the overarching threats from climate change and human activities. This final chapter explores how aquatic humans would fit into or disrupt existing marine food webs and what their presence would mean for ocean ecosystems. New species' ecological role begins with determining trophic level positioning within the marine food web. Trophic levels describe an organism's position in the energy flow from primary producers through herbivores to various levels of carnivores. Aquatic humans would almost certainly occupy a high trophic level, functioning as apex or subapex predators. Our terrestrial ancestors evolved as omnivores, but increasingly specialized as hunters of large prey. This predatory tendency would likely persist or intensify in aquatic environments where plant matter is less accessible than in terrestrial habitats. Predator or prey status would depend on body size and defensive capabilities. During early evolutionary stages, when aquatic humans remained relatively small and lacked specialized adaptations, they might be vulnerable to predation by large sharks, killer whales, and other apex predators. However, as body size increased, swimming speed improved, and group coordination became more sophisticated, aquatic humans would shift from potential prey to effective predators. Intelligence and social cooperation would provide advantages unavailable to most marine predators, potentially elevating aquatic humans to apex predator status despite lacking the massive body size of the largest marine mammals. Competition with existing marine mammals would create ecological tensions across multiple dimensions. Overlap with dolphin niches appears particularly likely given similar body sizes, social structures, and hunting strategies. Dolphins occupy diverse ecological roles from coastal fish hunters to deep-diving squid specialists. Aquatic humans would likely compete most directly with dolphin species exploiting similar prey in similar habitats. This competition could lead to niche partitioning, where species specialize on different prey types or forage at different times or depths, or to competitive exclusion, where one species drives the other from contested habitats. Competition with pinnipeds would occur primarily in coastal and continental shelf environments. Seals and sea lions hunt fish, squid, and invertebrates in relatively shallow waters, often hauling out on beaches or ice for rest and reproduction. During early evolutionary stages, aquatic humans maintaining semi-aquatic lifestyles would compete most intensely with pinnipeds for both food resources and haul-out sites. However, as aquatic humans became increasingly committed to fully aquatic existence, this competition would decrease as the species occupied increasingly different niches. Conflict with orcas represents perhaps the most significant competitive challenge. Killer whales are the ocean's apex predators, hunting everything from fish to the largest whales. Their sophisticated social coordination, diverse hunting strategies, and extreme intelligence make them formidable competitors. Aquatic humans and orcas would likely develop tense relationships characterized by competition for prey and potentially direct antagonism. Whether these species would coexist through niche partitioning, engage in active conflict, or develop unexpected cooperative interactions remains uncertain, but would fundamentally shape aquatic human ecology. Prey species impact would ripple through marine food webs as a new predator exploited existing resources. Overfishing concerns would arise if aquatic human populations grew large relative to prey availability. The carrying capacity of the seas and oceans for future sustainable exploitation has limits. Carrying capacity in the context of marine ecosystems refers to the maximum population of fish that an ecosystem can support without degradation. Studies of marine fisheries consistently show that exceeding carrying capacity leads to population collapses and ecosystem disruption. Carrying capacity is the stable number of individuals in a community, with limiting factors including living space, food availability, and physical and biological factors. For example, prey and predator populations have limits within a geographic area. If aquatic human populations expanded beyond what prey resources could sustain, cascading ecological effects would result. Declining fish populations would affect other predators competing for the same prey, potentially leading to starvation and population crashes in dolphins, seals, seabirds, and other marine species. Ecosystem balance disruption through removal of key prey species could trigger trophic cascades. Trophic cascades occur when removing predators or prey from one level affects populations at other levels, cascading through the food web. If aquatic humans preferentially hunted certain species, perhaps larger, slower fish that are easier to catch, these populations would decline, while competitors or prey of those species might increase, fundamentally altering community structure. Trophic cascade effects have been documented extensively in marine systems. The classic example involves sea otters, which prey on sea urchins, which graze kelp. When sea otters are removed, sea urchin populations explode and overgraze kelp forests, transforming diverse kelp ecosystems into barren sea urchin-dominated deserts. Similar cascades could result from aquatic human hunting patterns, though the specific effects would depend on which species are targeted and how heavily they are exploited. Habitat requirements for aquatic humans would encompass multiple environment types depending on their ecological specialization. Coastal versus open ocean preferences would separate populations into distinct ecotypes. Coastal populations would exploit productive continental shelf waters, rocky reefs, kelp forests, and estuarine environments. Open ocean populations would range across pelagic zones, diving deep for squid and fish far from land. These ecotypes might eventually diverge into separate subspecies or species through isolation and adaptation to different environments. Depth range preferences would vary with physiological capabilities and prey distributions. Shallow water specialists might forage primarily in the photic zone above 200 meters, where light penetrates and productivity is highest. Deep diving specialists might regularly descend to 500 to 1,000 meters or beyond, accessing mesopelagic and bathypelagic prey unavailable to shallow divers. Different depth preferences would reduce competition among aquatic human populations while allowing exploitation of diverse resources. Temperature tolerance zones would determine geographic ranges. Marine mammals show varying temperature preferences from Arctic and Antarctic specialists to tropical species. Aquatic humans would need to evolve cold tolerance for polar waters or remain restricted to temperate and tropical seas. Blubber thickness, metabolic rate, and behavioral thermoregulation would determine which temperature ranges each population could tolerate. Seasonal migrations between warmer and cooler waters might allow exploitation of temperature-dependent resources while avoiding thermal extremes. Breeding ground needs would require specific habitat features. Warm, shallow, protected waters would likely be preferred for giving birth and raising young, similar to patterns in many whales and dolphins. Specific bays, lagoons, or island systems might become traditional breeding grounds maintained through cultural transmission across generations. Protecting these critical habitats would be essential for population persistence. Geographic distribution potential spans the world's oceans, but with constraints. Ocean temperature limitations would exclude very cold polar waters unless extreme cold adaptations evolved. Food availability constraints would limit populations in oligotrophic open ocean regions where productivity is low. The most productive environments, including coastal upwelling zones, continental shelves, and areas with complex topography supporting diverse prey, would support the highest aquatic human densities. Migration route establishment would connect foraging grounds, breeding areas, and seasonal resources. Like many marine mammals, aquatic humans would likely migrate hundreds or thousands of kilometers annually, following prey distributions and environmental cycles. Traditional migration routes would be culturally transmitted, with experienced individuals guiding others along established pathways. Population dynamics would be governed by birth rates, death rates, and carrying capacity. Carrying capacity of oceans for large predatory mammals is limited by prey availability and habitat extent. Growth rate projections for aquatic human populations would depend on reproductive rates and survival to reproductive age. Marine mammals generally have low reproductive rates compared to terrestrial mammals, with extended gestation, long lactation periods, and multi-year intervals between offspring. Aquatic humans would likely show similar patterns, growing slowly but potentially living long lives. Mortality factors beyond predation would include starvation during periods of low prey availability, disease and parasitism, extreme weather events including storms and temperature fluctuations, injuries from aggressive interactions with conspecifics or other species, entanglement in debris, and anthropogenic causes if terrestrial humans persist. Disease susceptibility in marine mammals is already a significant conservation concern. Emerging viruses in marine mammals are impacting populations worldwide. Among the viruses recently isolated from marine mammals are influenza viruses, morbilliviruses, papillomaviruses, herpes viruses, arboviruses, and caliciviruses. These viruses may be associated with neoplasia, epizootics, and zoonotic disease, and involve complex pathogenesis involving non-infectious cofactors such as anthropogenic toxins, biotoxins, immunologic suppression, and other environmental stressors. Parasites specific to marine mammals include various nematodes, cestodes, and ectoparasites adapted to aquatic hosts. Aquatic humans would be exposed to these parasites, requiring immune system adaptations to manage infections. Bacterial infections in aquatic environments pose challenges as many bacteria thrive in seawater and can infect wounds. Viral transmission in water occurs readily in dense populations, potentially spreading disease rapidly through pods. The close social contact necessary for cooperative hunting and childcare would facilitate disease transmission, selecting for robust immune responses. Climate change impacts loom as perhaps the greatest threat to all marine life, including hypothetical aquatic humans. Ocean acidification effects result from increased atmospheric carbon dioxide dissolving in seawater, lowering pH and altering carbonate chemistry. Ocean acidification is fundamentally changing the chemistry of the world's oceans and threatening marine resources. The impacts of ocean acidification on marine organisms are widespread and significant. When all taxa are pooled together, ocean acidification has significant negative effects on survival, calcification, growth, development, and abundance. Survival and calcification are the responses most affected by acidification, with 27% reductions in both responses, whereas growth and development are reduced by approximately 11 to 19% for conditions roughly representing year 2100 scenarios. On average, abundance is reduced 15%. While aquatic humans themselves would not calcify shells acidification would devastate their prey. Many fish and invertebrates rely on calcified structures, and their decline would cascade through food webs, reducing prey availability for aquatic humans. Temperature changes and range shifts would force aquatic humans to adapt to warming oceans. Rising temperatures alter species distributions, pushing marine life toward poles and into deeper, cooler waters. Aquatic humans would need to either migrate with shifting prey distributions or adapt physiologically to warmer conditions. Food web alterations from climate change include shifts in plankton communities, affecting all higher trophic levels. Changes to ocean currents and stratification patterns affect nutrient upwelling and productivity. Disruption of seasonal cycles affects breeding and migration timing, potentially causing mismatches between predators and prey. These complex changes create an uncertain future for all marine species. Pollution affects compound climate impacts. Ocean plastic pollution affects over 900 megafaunal species, of which more than 100 are endangered. Entanglement and ingestion of plastic debris cause injury and death. Filter-feeding baleen whales are especially vulnerable to microplastic ingestion. These plastics attract toxic chemicals known as persistent organic pollutants from the water, and once consumed, these toxins accumulate in tissues such as blubber. Species high up in the food chain, including marine mammals, are especially vulnerable to biomagnification. By feeding on prey that has also ingested plastic, they accumulate ever higher levels of harmful chemicals. This toxic buildup disrupts endocrine and reproductive systems, further endangering survival. Aquatic humans would face identical risks, accumulating toxins through the food chain with potentially severe health consequences. Human-modified aquatic-human conflicts would arise from interactions with terrestrial human activities. Fishing industry interactions present the most immediate threats. Commercial fishing operations are the largest cause of human-related injury and death for many marine mammals. Bycatch is the #1 source of direct human-caused death and serious injury to marine mammals worldwide, estimated at over 650,000 individuals each year. The primary human-induced cause of mortality of marine mammals is incidental capture and entanglement in fishing. Aquatic humans would face identical bycatch risks if commercial fishing persists. Entanglement in gillnets, trawls, and other gear could cause drowning, strangulation, or severe injuries. Shipping lane dangers would increase as aquatic humans occupied the same waters traversed by cargo ships, tankers, and other vessels. Ship strikes cause significant mortality in large whales and would pose similar threats to aquatic humans. High-speed vessels in particular present collision risks, especially in areas where shipping lanes cross migration routes or important foraging grounds. Conservation considerations for aquatic humans would mirror those for other marine mammals. Protected status needs would require legal frameworks designating aquatic humans as protected species, prohibiting hunting and harassment. Habitat preservation would require establishing marine protected areas encompassing critical breeding grounds, foraging areas, and migration corridors. These protected areas would need adequate size and connectivity to support viable populations. Genetic diversity maintenance would require preserving multiple distinct populations to maintain evolutionary potential and reduce inbreeding risks. The emergence of aquatic humans would represent a profound transformation not just of our species, but of entire ocean ecosystems. The ecological impacts would be far-reaching, complex, and difficult to predict with certainty. Competition, predation, disease dynamics, and environmental stresses would all shape whether and how aquatic humans could successfully persist in marine environments. Success would require not just biological adaptations, but also sustainable population sizes, respecting ocean carrying capacity, harmonious coexistence with existing marine species, and resilience in the face of anthropogenic environmental changes. The journey from terrestrial to aquatic life is possible, as cetaceans, pinnipeds, and sirenians have conclusively demonstrated. For humans to make that same journey would require millions of years of evolutionary change, transforming virtually every body system while navigating immense ecological challenges. The resulting species would be radically different from modern humans, adapted as thoroughly to ocean life as whales and dolphins are today. They would swim through three-dimensional spaces with powerful flukes, hunt with echolocation or keen underwater vision, give birth tail first to precocial young, sleep with one brain hemisphere awake, and communicate through complex underwater vocalizations. They would be, in every meaningful sense, marine mammals, retaining perhaps fragments of their terrestrial heritage, but fully committed to the ocean. Whether such evolution will ever actually occur remains unknown and unknowable. It would require selection pressures intense and sustained enough to drive populations increasingly toward aquatic specialization across thousands of generations. It would require that humans survive long enough as a species for such long-term evolution to unfold. And it would require that the oceans themselves remain viable habitats, not degraded beyond recovery by pollution, climate change, overfishing, and habitat destruction. The future is uncertain, but the past provides clear evidence that the transformation from land to sea, while requiring millions of years and countless small steps, is entirely possible for mammals. The blueprint exists in fossil whales and living dolphins. The question is not whether it could happen, but whether circumstances will ever favor such a dramatic evolutionary journey for humanity. If you found this exploration of aquatic human evolution fascinating, make sure to like this video and subscribe to the channel for more deep dives into evolutionary biology, marine science, and the incredible diversity of life on Earth. Share your thoughts in the comments. Do you think humans could ever actually evolve to become aquatic? What adaptations do you think would be most challenging? What other evolutionary scenarios would you like us to explore? Thanks for watching, and we will see you in the next video.